Pipe dream, aquaculture cities.
Dream Part 5. The Engineered Ecosystem. MXTM aka Pirate 1 or VJ Tsunami X. July 11, 2026. Chapter 1. Cleaner Fish. The cleaning station is one of ecology's most enduring institutions. On every coral reef in every tropical ocean, at specific locations whose positions are learned and remembered by the reef's fish populations across generations, a small fish waits. Larger fish arrive. They adopt a posture, fins spread, mouth open, gill covers flared, that signals, I am not threatening, I am not fleeing, I am presenting myself for service. The cleaner removes ectoparasites, dead tissue, and accumulated debris from the client's skin, gills, and oral cavity. The client holds still and does not eat the cleaner. The cleaner eats and does not flee the client. The exchange is mutual benefit organized by behavioral signal, maintained by evolutionary pressure over millions of years on both participants. The cleaning station is a biological institution that predates human institutions by geological margins. It does not require enforcement. It does not require contract. It does not require the client fish to remember that this particular cleaner provided good service last Tuesday. It requires only that the behavioral signals are read correctly by both parties, that the presenting posture triggers the cleaner's approach response, and that the cleaner's color pattern and movement signature triggers the client's tolerance response. The institution is in the signals. The signals are in the organisms. The organisms are in the ecology. Pipe Dream's crystal tube network is a reef without a cleaning station. The tube walls, polycarbonate in the halocline zone, cast PMMA at the surface tier, chemically strengthened aluminosilicate glass at structural depth, are substrate. Substrate in a cenote water column is not unoccupied. It is colonized. The colonization begins with the conditioning film, the adsorbed layer of dissolved organic molecules that the water chemistry deposits on any new surface within hours of installation, and proceeds through the bacterial adhesion, the biofilm succession, the mature fouling community, on the schedule the passage water's biological seed stock and the tube wall surface chemistry determine. The tube wall fouling problem is not a problem the civilization introduced by installing the crystal tube. It is the formation's biological reality applied to the new substrate the civilization provided. The cenote's biological community does not distinguish between a limestone wall and a polycarbonate tube wall at the scale of the individual bacterium. Both are surfaces in nutrient-containing water. Both will be colonized. The fouling is certain. The question is only whether the civilization manages it or is managed by it. The cleaning station the crystal tube network requires is not the coral reef's cleaning station. The tube wall's substrate is not coral. The tube wall's biological community is not the reef's parasite and debris-laden fish skin. The cleaning function that the crystal tube requires is the dissolution of biofilm base adhesive, the extracellular polymer matrix that anchors the biofilm community to the polycarbonate surface, at the specific chemistry of that surface, in the specific water conditions of the specific depth zone the tube occupies, at the spatial coverage and temporal frequency that the crystal specification's optical transmission threshold requires. No natural cleaning station organism performs this function. The loricariid catfishes of the surface world's freshwater ecosystems scrape algae and biofilm from hard surfaces. They are herbivores and detritivores whose feeding appendages are optimized for mechanical scraping of substrate-attached biological material. Their enzymatic biology is not optimized for the polycarbonate surface chemistry of an engineered underwater structure. Their behavioral ecology is not organized around a cleaning station institution of the kind the crystal tube network requires. Their cleaning behavior serves their nutrition, not the substrate's optical clarity. The Ancestris vitreus was designed from this starting point, a loricariid catfish as the biological chassis, because the loricariid's oral disc morphology, benthic feeding behavior, and freshwater adaptation provide the correct physical foundation, and a set of modifications to the enzymatic biology, the skin's optical properties, the lateral line's communication interface, and the behavioral response to the coordinating system's acoustic management signals that convert the loricariid chassis into the crystal tube's primary maintenance organism. The natural organism provided the body plan. The design provided the specific capabilities the body plan was not naturally equipped with. The oral disc's enzymatic chemistry. The loricariid catfish's oral disc is an evolutionary masterpiece of adhesion biology. The disc's muscular rim creates suction against hard substrates, sufficient adhesive force to maintain position on smooth rock surfaces against substantial water current. The disc's interior surface carries the rasping structures, modified teeth and papillae, that the natural loricariid uses to graze algae and biofilm from the substrate. In the natural organism, the rasping is mechanical, the papillae physically remove attached biological material by abrasion and the oral suction holds the fish in position while the rasping proceeds. The Ancestris vitreus's oral disc retains the suction mechanism and the position-holding function but replaces the mechanical rasping biology with a chemical mechanism. The modified papillae do not rasp. They secrete. The secretory apparatus, glandular tissue distributed across the oral disc's inner surface, supplied by ducting from the posterior secretory glands. The design specification introduced to the lower carried chassis body plan, produces the enzymatic mucus that the crystal specification's maintenance function requires. The enzymatic mucus's active component is a polysaccharide lyase, an enzyme class that cleaves the glycosidic bonds in the extracellular polysaccharide chains that constitute biofilm's primary adhesive matrix. The polysaccharide lyase does not attack the bacterial cells themselves. It attacks the molecular scaffold that anchors the bacterial cells to the substrate surface. The biofilm's structural integrity depends on this scaffold. Remove the scaffold's continuity and the biofilm loses its mechanical connection to the substrate surface. The bacterial cells and the matrix fragments that the scaffold's cleavage produces become suspended material in the water column, removed from the surface by the passage's normal water flow. The specificity of the lyase for polysaccharide scaffolds rather than for the bacterial cells themselves is the design critical property. A cleaning mechanism that kills bacteria, through biocidal chemistry or physical disruption, leaves bacterial cell debris on the substrate surface and releases bacterial intracellular content into the water column. The cell debris and intracellular content are nutrients for the next colonizing community. The surface after biocidal cleaning carries a nutritional legacy that accelerates recolonization by the next biofilm community's pioneer species. The cleaning creates the conditions for the next fouling. The polysaccharide lyase mechanism avoids this. The scaffold is cleaved, the cells are removed with the scaffold fragments by water flow, and the substrate surface is left without the conditioning film chemistry that the biofilm's polysaccharide matrix modifies to favor recolonization. The surface after enzymatic cleaning is not the biocide-treated surface carrying bacterial debris. It is the bare substrate surface, polycarbonate, PMMA, or aluminosilicate glass, whose surface chemistry is the same as the day the crystal tube section was installed, before any conditioning film had established. The recolonization timeline from this surface is the timeline from zero conditioning film density, the longest possible interval between cleaning cycles, because the conditioning film must re-establish before bacterial adhesion can proceed, and bacterial adhesion must establish before the biofilm community's succession can begin. The enzymatic mechanism resets the surface's biological age rather than simply removing the current community. This is the key distinction. The mechanical cleaning of the natural lower caryoids rasping removes the mature community but leaves the conditioning film and the earliest bacterial adhesion products that the rasping could not access at the molecular scale. The enzymatic cleaning removes the adhesive scaffold at the molecular scale, leaving the substrate surface in a state closest to its installation day chemistry. Each cleaning cycle gives the surface the longest possible period before the next cleaning cycle is required. The polysaccharide lyase's substrate specificity, its activity against the polysaccharide scaffolds that the specific bacterial community colonizing the crystal tube walls in the Yucatan's freshwater aquifer produces, was optimized through a directed evolution protocol applied to the lyase's binding domain during the Ancestris vitreous's development phase. The binding domain determines which molecular structures the enzyme cleaves. The directed evolution protocol selected for binding domain variants that maximized cleavage efficiency against the polysaccharide scaffolds the cenotes bacterial community actually produces, as characterized by the Hydroprint campaign's microbiological surveys of the founding cenotes biological community composition. The enzyme is tuned to the cenote. Not to a generic biofilm. Not to a laboratory bacterial culture. To the specific molecular architecture of the adhesive scaffolds produced by the specific bacterial species that colonize the specific substrate chemistry of crystal tube wall materials in the specific water chemistry of the Yucatan's freshwater aquifer. The tuning required the Hydroprint campaign's biological census data before any enzyme design could be finalized. The cleaning biology was specified after the formation was characterized, not before. The organism is an answer to the formation-specific biological question. The chromatophore skin. The natural lower caryoid catfish is not a beautiful fish. It is an armored, bottom-dwelling, nocturnal feeder whose evolutionary pressures optimized for predator avoidance, suction adhesion, and nocturnal foraging rather than for any aesthetic criterion. Its coloration is typically drab, brown, gray, or black with variable patterning, and its skin's optical properties reflect its benthic ecology. An organism that lives pressed against rocky substrates in murky rivers does not need the optical spectacle of a reef fish. The Ancestris vitreous's skin received the modification that the crystal tube's optical specification demanded, the introduction of chromatophore cells derived from cephalopod photoreceptor biology into the skin's outer layers. Cephalopod chromatophores are among biology's most sophisticated optical control mechanisms. The cuttlefish and the octopus can change their skin's color, pattern, and texture, including its apparent three-dimensional relief, in milliseconds, through a system of chromatophore cells controlled by direct neural innervation. The color change is not a hormonal response requiring minutes to manifest. It is a neural response requiring milliseconds. The chromatophore cell's muscular sheath contracts or relaxes under direct nerve stimulation, expanding or contracting the pigment sac at the cell center, changing the surface area of pigmented cell that the observer sees and thus changing the skin's apparent color in that location. The Ancestris vitreus carries chromatophores derived from this cephalopod biology in its skin's outer layer, with a specific modification to the pigment sac's content, rather than the varied pigment types that cephalopod chromatophores use to produce the full visible spectrum color range. The Ancestris vitreus's chromatophores contain a pigment whose optical properties in the relaxed, pigment sac fully open, large pigmented area visible state produce the organism's standard coloration. Visible to the observer as a distinct biological object on the tube wall surface, and in the contracted, pigment sac minimized, minimal pigmented area visible state approach optical translucency. The translucent state is the organism's resting state on the tube wall surface during an active cleaning session. When the Ancestris vitreus has secured its oral disc suction on the tube wall and begun the enzymatic secretion cycle that characterizes active cleaning, the neural pathway that controls the chromatophore contraction is activated simultaneously with the oral disc's secretory apparatus. The organism cleaning the wall contracts its chromatophores. The organism whose chromatophores are contracted is semi-translucent. The semi-translucent organism on the tube wall surface does not read as an opaque biological mass interrupting the visual field through the tube wall. It reads as a faint shimmer, a visual perturbation less obstructive than the biofilm the organism is removing. The guest in a crystal tube pod transiting through a section where the Ancestris vitreus colony is actively cleaning sees the limestone formation visible through the tube wall, the cenote water column between the tube exterior and the formation, and at the tube wall surface, the faint shimmering presence of the cleaning colonies, visible not as fish, not as opaque masses, but as a barely there optical effect at the tube wall surface that the observer knows as organisms if they know what to look for, but that does not interrupt the formation's visual continuity if they are not specifically attending to it. The organism's translucency during cleaning is not camouflage. The formation observer can see the Ancestris vitreus if they look for it. The translucency is a calibrated optical contribution to the crystal specification's maintenance. The cleaning organism does not compromise the optical standard it is maintaining. The organism on the wall is part of the visual field through the wall, but a visually minor part, less visually disruptive than the biofilm it is removing would be if left unaddressed. When the organism releases its oral disc suction and moves to the next cleaning position, when the chromatophore contraction is released and the organism is in transit between cleaning positions, the organism becomes visible at full coloration. The transit phase lasts seconds. The cleaning phase, which triggers the translucency, lasts minutes to hours per location depending on the fouling load. The observer who notices the Ancestris vitreus colony is most likely to notice it during the transit phase, when the organisms are visible at full coloration moving across the tube wall. The cleaning phase is visible only to an observer who knows the translucent shimmer is an organism performing a maintenance function. The lateral line as management interface. The fish's lateral line system is among vertebrate biology's most sophisticated sensory innovations, a distributed mechanosensory array running along the fish's body from head to tail, detecting pressure gradients, water velocity changes, and low-frequency vibration in the water column. The lateral line is the fish's primary non-visual environmental sensor, the system through which it detects approaching predators, schooling companions, and the hydrodynamic signatures of prey organisms. In the dark, turbid waters of the lower Cariad's natural environment, the lateral line is the primary sense. The Ancestris vitreus's lateral line retains its natural mechanosensory function. The fish uses it to navigate in the cenote's water column, detect the acoustic management signals the coordinating system's ultrasonic pings produce, and maintain its spatial relationship to other members of the cleaning school during transit between cleaning zones. The biointegrated microtransponder tag introduced to the lateral line's canal system adds a communication function the natural lateral line does not have. The transponder receives the coordinating system's ultrasonic management pulses and translates them into the electrical signals that the lateral line's native neural pathway interprets as directional cues. The coordinating system does not transmit navigation instructions to the Ancestris vitreus in the sense that it transmits position instructions to the swarm unit. The swarm unit receives a position offset and moves by the offset. The Ancestris vitreus receives an ultrasonic signal that its lateral line interprets as an acoustic stimulus from a specific direction and at a specific intensity, triggering the fish's natural behavioral response to lateral line stimulation, movement toward the signal source. The coordinating system steers the cleaning school by placing the ultrasonic signal source, the transponder network's acoustic output, in the direction of the optical transmission anomaly the coordinating system's sensor network has identified as requiring cleaning attention. The fish follows the signal. The signal comes from the direction of the fouling zone. The fish arrives at the fouling zone. The oral disc's chemosensory detection of the elevated dissolved organic carbon concentration that the mature biofilm releases into the surrounding water column confirms the fouling zone's location and triggers the cleaning behavior's initiation, the oral disc suction engagement, the enzymatic secretion commencement, the chromatophore contraction, the stationary feeding position. The management interface is not mind control. The coordinating system does not override the fish's behavioral responses. It places acoustic stimuli that trigger the fish's natural responses, approach toward acoustic stimulus source, feeding behavior initiation at elevated organic carbon concentration, stationary feeding posture at suction disc engagement. The fish does what a loricariid does in response to its sensory environment. The coordinating system manages the sensory environment to produce the cleaning coverage the crystal specification requires. This is biological management rather than biological command. The coordinating system manages the conditions to which the organism responds, not the organism's responses to conditions. The distinction has a practical consequence for the management system's resilience. An organism whose responses are commanded by an external signal fails when the command signal fails. An organism whose natural responses are triggered by managed conditions fails only when the conditions fail, and the conditions the coordinating system manages are acoustic signals, which the acoustic transponder network produces from the streaming potential harvest system's power output at the freshwater zone's depth. The power source for the management signal is the formation's own chemistry. The management signal is available as long as the formation's chemistry produces the streaming potential. The organism responds as long as the management signal is available. The cleaning coverage is maintained as long as the formation is hydrating. The school as maintenance unit. Individual ancistrous vitreous organisms do not clean crystal tube wall sections. Schools do. The cleaning school is the management system's operative unit, the aggregate of organisms that the coordinating system's acoustic management deploys to a fouling zone and that collectively cover the zone's wall surface in the residence time the optical transmission decline rate allows before the zone crosses the crystal specification threshold. The school's spatial organization during active cleaning is not the typical fish school's formation, the tight, coordinated movement of many individuals maintaining precise spacing while changing direction together that the surface world observes in open water schooling species. The Ancistrus vitreous's cleaning school is distributed across the wall surface, individuals at different positions on the wall, each attached by oral disc suction, each cleaning the surface in the immediate vicinity of its attachment point, with the school's coverage density determining the area cleaned per unit time. The coverage density is the management variable the coordinating system controls through the acoustic management signal's intensity. Higher signal intensity attracts more individuals from adjacent non-fouled zones to the target zone, increasing the school's local density. Lower signal intensity allows individuals to disperse to adjacent zones as the local fouling is addressed. The signal intensity is proportional to the optical transmission deviation from the crystal specification threshold. Higher deviation, higher intensity, larger school, faster coverage. The school size self-adjusts to the fouling severity without requiring the coordinating system to specify a school size target. The coordinating system specifies the signal intensity. The school size responds to the signal intensity through the individual organism's natural attraction response. The cleaning coverage rate is the product of the school size and the individual organism's cleaning rate. All three parameters are management outputs from the single control variable, the acoustic signal intensity at the target zone. The school's collective behavior also produces a maintenance byproduct that no individual organism produces, the hydrodynamic disturbance generated by many organisms moving across the wall surface in the same general direction stirs the boundary layer, the thin layer of still water immediately adjacent to the wall surface, that would otherwise protect the wall surface from the water column's flow and allow the biofilm community's waste products to accumulate at the wall surface, accelerating the conditioning film's re-establishment after a cleaning cycle. The school's movement-induced boundary layer disturbance reduces the conditioning film's re-establishment rate, extending the interval between cleaning cycles beyond what the enzymatic cleaning alone would produce. The boundary layer disturbance is not a designed function. It is an emergent hydrodynamic effect of multiple organisms moving in the same general direction in proximity to a wall surface. The coordinating system did not specify it. The fish biology produces it as a consequence of the school's cleaning behavior. The cleaning coverage maintenance benefit, the extended interval before recolonization that the boundary layer disturbance contributes to, is the formation's bonus. The ecology produces more than the specification required. The school's social structure also contributes to the cleaning coverage's spatial distribution in a way the coordinating system does not manage. Individual Ancistrus vitreous organisms do not clean the same surface location twice in immediate succession. After completing an enzymatic cleaning session at one location, the individual moves to a new location rather than returning to the just cleaned location. This behavioral tendency, which the loricariid chassis' natural foraging behavior produces, since herbivorous grazers do not return to a location they have just depleted, distributes the school's cleaning effort across the wall surface rather than concentrating it at the most fouled point that attracted the school's initial response. The distribution is not coordinated by the coordinating system. It is the natural outcome of individual foraging behavior applied to a wall cleaning context. The school's collective coverage is more spatially efficient than the sum of individually optimized cleaning visits would be. The halocline limit. The Ancistrus vitreous cannot cross the halocline. This is not a management restriction the coordinating system enforces. It is a biological limit the organism's physiology imposes on itself. The halocline's salinity gradient and the increasing H2S concentration below the halocline produce physiological conditions the Ancistrus vitreous's osmoregulatory biology cannot manage. The organism's kidneys and gills, specified for freshwater ion regulation, cannot adapt rapidly enough to the salinity change the halocline imposes to maintain the blood chemistry the organism's neural function requires. The H2S chemistry that begins below the halocline interferes with the cytochrome oxidase pathway that the organism's aerobic metabolism depends on. Below the halocline, the ancestris vitreous dies. This limit is the most consequential design constraint the ancestris vitreous carries, and it was known before the first organism was deployed. The crystal tube network extends below the halocline. The tube walls below the halocline foul on the same schedule as the tube walls above it. The biology differs by depth zone, but fouling biology is present at every depth. The ancestris vitreous can maintain the crystal specification in the freshwater zone. It cannot maintain the crystal specification in the halocline zone and below. The squeegeebot fleet is the mechanical solution to this biological limit. The squeegeebot's flat disk geometry, neodymium magnetic roller clamping system, and ultrasonic cavitating blade maintenance mechanism were designed specifically for the depth zones the ancestris vitreous cannot enter. The squeegeebot does not perform the enzymatic base adhesive dissolution that the ancestris vitreous produces. It performs physical cavitation disruption of the biofilm at the cellular detachment scale, the ultrasonic frequency and intensity calibrated to the cavitation energy that detaches biofilm cells from the substrate surface without scratching the optical finish, 6 orders of magnitude different in energy from the lithocrustacean's limestone fracture cavitation. The squeegeebot's physical mechanism is less effective at preventing recolonization than the ancestris vitreous's enzymatic mechanism. The squeegeebot removes the biofilm community but does not remove the conditioning film at the molecular scale the lyase achieves. The interval between squeegeebot cleaning cycles is shorter than the interval between ancestris vitreous cleaning cycles for equivalent fouling rates. The coordinating system's maintenance scheduling allocates squeegeebot coverage at higher frequency than ancestris vitreous coverage would be allocated for the same tube section if the biological cleaning option were available at the halocline zone's depth. The halocline limit and the squeegeebot response to it are the biological management system's most legible boundary. Here, biology; below, mechanics. The boundary is chemical and biological, the halocline's chemistry, rather than administrative. No protocol specifies that the ancestris vitreous will not cross the halocline. The halocline specifies it. The management system respects a limit the formation imposed. Population dynamics in a managed ecosystem. The ancestris vitreous population in the founding formation cenotes freshwater zone is not a fixed resource the coordinating system deploys and manages. It is a living population whose size, age structure, spatial distribution, and genetic composition change continuously through the biological processes the managed habitat supports: birth, growth, death, movement, and the genetic drift the founding biologists specified the monitoring program to address. The population size dynamics are managed through the habitat conditions the coordinating system controls rather than through direct demographic interventions. The freshwater zone's food web supports the ancestris vitreous through the biofilm and algae communities that colonize the crystal tube walls and the cenote's natural limestone surfaces. The same communities the cleaning school removes from the tube walls are available on the natural limestone surfaces as the population's supplementary food source, not depleted by cleaning activity because the natural limestone surfaces are not the target of the acoustic management signal's cleaning deployment. The coordinating system manages the food web's productivity through the light relay spectral output. The photosynthetically active radiation that the light relay delivers to the freshwater zone's water column at the wavelengths that algal photosynthesis uses most efficiently determines the algal community's primary productivity. Higher photosynthetically active radiation increases algal productivity, increases the biofilm community's growth rate on all surfaces in the freshwater zone, increases the food availability for the ancestris vitreous population, and increases the population's reproductive rate and survival rate. Lower photosynthetically active radiation reduces the food web's productivity and reduces the population's growth rate. The coordinating system's population census, weekly individual counts by the biological monitoring network's optical sensors, interpreted by the coordinating system's population model, provides the current population size estimate. The model projects the population's growth trajectory from the current census against the food web's current productivity and the habitat's current carrying capacity. If the projection indicates that the population will exceed the carrying capacity within the planning horizon, the coordinating system reduces the light relay's photosynthetically active output, reducing food web productivity, slowing the population's growth rate toward the carrying capacity's sustainable level. If the projection indicates that the population is declining below the level the crystal specification's cleaning coverage requires, the coordinating system increases the photosynthetically active output. The population is managed through its food web. The food web is managed through the light relay. The light relay is managed by the coordinating system. The ancestris vitreous population size is a managed state variable, not a fixed infrastructure component. Its size changes with the cleaning demand the crystal specification creates and the habitat capacity the coordinating system maintains. The management is indirect, through the trophic chain that connects the light relay to the algal community to the biofilm food web to the ancestris vitreous's feeding behavior, but the connection is tight enough for the coordinating system's population model to maintain the cleaning coverage the crystal specification requires. The genetic drift monitoring program tracks the population's genetic composition at the founding specification's critical loci, the enzymatic lyases binding domain, the chromatophores contraction pathway, the lateral lines transponder response sensitivity, the osmoregulatory biology's halocline sensitivity threshold. The last locus, the halocline sensitivity threshold, carries particular monitoring priority. A drift event that increased the population's tolerance for the halocline salinity gradient would expand the individual's depth range into the halocline zone. Individuals crossing the halocline would encounter the H2S gradient and die before reaching the full halocline zone's concentrations, but their entry into the halocline zone would create a biological corridor exposure event that the Biological Corridor Management Protocol's pre-transit inspection protocol is designed to prevent through other means. Individuals dying at the halocline boundary would release biological material into the halocline zone that the halocline zone's water chemistry monitoring would detect, and the coordinating system would flag as a biological anomaly requiring terraform operator review. The monitoring program's detection of the halocline sensitivity locus's drift toward increased tolerance triggers genetic archive introduction before any individual crosses the halocline. The correction is at the population genetics level, before any behavioral boundary crossing occurs, because the genetic monitoring's resolution is finer than the behavioral monitoring's resolution for this specific risk. The genetic signature of halocline sensitivity drift appears in the population's allele frequencies before any individual organism manifests the behavioral consequence of the drift. The cleaning station institution. The cleaning station's evolutionary history on the coral reef produced an institution whose participants have no explicit knowledge of the institution's existence. The cleaner fish does not know it is a cleaning station. The client fish does not know it is visiting a cleaning station. Each responds to the behavioral signals the other produces, the presenting posture, the cleaner's color pattern, the tactile stimulation of the cleaning behavior, and the institution persists because the individual responses produce mutual benefit that evolutionary pressure has stabilized. Pipe Dream's cleaning institution is organized differently. The Ancestris vitreous is directed to specific cleaning locations by the coordinating system's acoustic management rather than by the chemical gradients and behavioral signals of a natural cleaning station ecology. The crystal tube wall is not a client that can present itself for service. The cleaning institution's organization is external. The coordinating system functions as the institution's organizing intelligence, replacing the evolutionary pressure that organized the reef's cleaning station with the management intelligence of the digital twin's optical transmission monitoring. But the institution that results has the same fundamental character as the reef's cleaning station: organisms performing a specific biological function, the removal of accumulated biological material from a surface, in exchange for the nutrition that the surface's biological community provides. The Ancestris vitreous eats the biofilm it removes. The tube wall provides the food. The tube wall's optical clarity is the exchange, not a benefit the tube wall receives in any meaningful sense, but a benefit the civilization receives from the organism's feeding behavior directed at the tube wall rather than at some other substrate. The institution differs from the reef's cleaning station in one critical respect: the reef's cleaning station is evolutionarily stable. The behaviors that constitute it have been selected for over geological time, are genetically encoded in both participants, and persist without management. Pipe Dream's cleaning institution is management stable. It persists as long as the acoustic management signal directs the Ancestris vitreous to the tube walls rather than to the natural limestone surfaces, where equivalent biofilm communities grow without the optical significance that the crystal specification assigns to the tube wall community. The natural limestone surfaces are an alternative food source the Ancestris vitreous is attracted to by the same chemical gradient signals, the dissolved organic carbon from the biofilm community, that attract it to the tube walls. Without the acoustic management signal's directional bias toward the tube walls, the Ancestris vitreous population would distribute its cleaning effort between the tube walls and the natural limestone surfaces in proportion to the food availability at each, which would not maintain the crystal specification's coverage requirement, because the natural limestone surface's food availability exceeds the tube wall's food availability in most freshwater zone passage sections. The natural limestone surfaces occupy more area, carry older and more productive biofilm communities, and are not maintained at the crystal specification's cleaning frequency that keeps the tube wall's biofilm communities at early succession. The tube walls are the less productive foraging habitat without management. The management signal makes the tube walls the more immediately rewarding habitat by directing the cleaning school to the tube wall locations with the highest fouling, the highest dissolved organic carbon concentrations, which the management signal's position tracks against the optical transmission monitoring. The cleaning institution's management stability requires the acoustic management signal's continuous operation. The management signal requires the acoustic transponder network's operation. The transponder network requires the streaming potential harvest system's power supply. The power supply requires the formation's underground river system to flow. The formation's underground river system flows as long as the Yucatan receives rainfall. The cleaning institution is stable as long as the cenote receives the rainfall that maintains the formation's hydrology. The institution's fundamental stability condition is the same as the civilization's fundamental habitat condition. The cleaning, coverage, maintenance, and the civilization's persistence are coupled at the hydrological base layer. A formation that cannot sustain the civilization cannot sustain the cleaning institution. A formation that sustains the civilization sustains the cleaning institution. The Ancestris vitreous's management stability is the civilization's management stability, expressed in the specific form of tube wall optical transmission maintenance. What the Ancestris vitreous knows. The Ancestris vitreous knows what its sensory system tells it: the acoustic signal's direction and intensity, the water column's chemical composition in the region of its current position, the tactile sensation of the tube wall surface against its oral disc, the boundary layer's hydrodynamic signature as other school members move nearby. It knows nothing of the crystal tube standard, the crystal specification's optical transmission threshold, the coordinating system's maintenance scheduling, or the civilization's architectural commitment to visual transparency as a governance principle. The organism is performing a biological function, eating biofilm from a hard surface, that the natural loricariid performs on natural limestone substrates throughout the freshwater systems of tropical South America. The surface is different. The mechanism is modified. The management context is entirely novel. But the organism's experience of its own activity is: there is a surface, there is food on the surface, the acoustic signal indicates that surface as the foraging destination, the oral disc is attached, the enzymatic secretion is active, the food is available. The organism eats. The civilization that designed the organism, manages the organism, and benefits from the organism's cleaning function has invested years of biological engineering work and decades of population management in a system whose operative agent has no knowledge of any of it. This is not unusual in engineering. The electron moving through the crystal tube's power conduit has no knowledge of the grid topology it is contributing to. What is unusual is that the operative agent is not a particle or a fluid but a living organism whose experiential state, whatever experience a fish has, is the state of an organism feeding and moving and schooling in a habitat that its biology finds suitable. The Ancestris vitreous is not suffering in service to the crystal specification. It is eating in a habitat where food is available, in a school where social cues are present, in water conditions its physiology supports. The management system has not reduced the organism to a tool, it has placed the organism in conditions where the organism's natural behaviors produce the management system's required outcomes. The organism is doing what its biology does. The biology doing what it does produces the crystal specification's maintenance. This alignment between what the organism does and what the management system needs is the deepest principle of biological civil engineering. The designed organism is not designed against its nature. It is designed so that its nature, the specific version of the organism's biology that the founding engineers specified, produces, in the cenote's conditions, the functions the civilization requires. The organism feeds. The tube wall is clean. The crystal specification is maintained. The civilization sees through the tube wall to the formation beyond it. The formation is visible because something is eating the film that would obscure it. The something is a fish. The fish is eating because it is alive and the food is there. The civilization designed the fish to find the food in the place the civilization needed the food removed from. The formation does not know this. The fish does not know this. The civilization built the relationship between the fish and the formation and benefits from the relationship without the relationship's other participants needing to know what they are part of. This is the quiet competence of biological civil engineering, arranging biology so that biological things doing biological things produce civil engineering outcomes. The engineering is in the arrangement. The doing is in the biology. The outcome belongs to both. Integration with the regional network. The Ancestris vitreous population management that the founding formation cenote's freshwater zone has developed across its operating period is the reference standard for every corridor cenote installation's biological management protocol. The regional network's biological management layer, the Network Governance Council's concurrent authority over cross-network biological standards, specifies the Ancestris vitreous's population management parameters as network-wide standards. The acoustic management signals intensity calibration against the optical transmission threshold, the food web productivity management through the light relay's photosynthetically active output, the genetic drift monitoring loci and correction response. Protocol, the school deployment scheduling against the coordinating system's optical transmission monitoring. Each corridor cenote installation receives the reference standards deployment package when its freshwater zone's biological corridor preparation is complete. The reference standard is not a rigid specification that every installation applies identically. It is a baseline from which each installation's terraform operator adjusts the management parameters to the specific freshwater zone's biological community composition, the specific passage network's hydrodynamic conditions, and the specific cenote's light cannon relay's achievable photosynthetically active output given the cenote opening's dimensions and orientation. The adjustments are the terraform operator's professional contribution to the biological management system, the translation of the reference standard's general parameters into the specific installation's operating conditions. The reference standard specifies the management objectives: cleaning coverage density, crystal specification compliance rate, population census frequency, genetic drift monitoring interval. The Terraform operator specifies the management parameters that achieve those objectives in this specific cenote. The regional network's collective biological management experience, the operational data from all cenote installations and Cistrus vitreous population management, accumulated across the coordinating system's network-wide data integration, feeds back into the reference standards revision at each annual biological management review. An installation whose adjustment to the reference standard produced superior cleaning coverage per unit of management signal power becomes a data point that the Network Governance Council's biological management review can incorporate into the next reference standard revision. The reference standard improves with the network's collective experience. The network's biological civil engineering capability improves as the reference standard improves. The improvement compounds with each annual revision. The Cistrus vitreous is the regional network's most widely deployed designed organism. Its population is present in every freshwater zone of every cenote installation in the network. Every crystal tube wall section in the freshwater zone in every cenote in the Principality is being maintained by organisms from the same designed biological chassis, managed by the same reference standard, monitored by the same genetic drift program, directed by the same coordinating system's acoustic management. The regional network's crystal specification is a uniform standard. The regional network's cleaning coverage is a distributed biological function. The distribution is the network's resilience. No single installation's cleaning colony failure affects the network's overall crystal specification compliance, because the crystal specification is maintained installation by installation, school by school, organism by organism, each performing its biological function in its specific habitat zone. The specification is uniform. The biology is distributed. The compliance is the product of distributed biology performing uniformly against a single standard. This is what biological civil engineering looks like at the regional network scale: a single standard, distributed biology, and the coordinating system managing the distribution's alignment with the standard at every installation simultaneously, updating the standard annually from the distribution's collective performance data, and producing a cleaning infrastructure that improves every year because the organisms maintaining it are alive, and living systems that are correctly managed improve rather than degrade. The Cistrus vitreous does not improve itself consciously, but the management system that manages it improves every year because the organisms maintaining it are alive, and living systems that are correctly managed improve rather than degrade. The Cistrus vitreous does not improve itself consciously, but the management system that manages it improves from the data the organism's performance produces. The organisms eat. The data accumulates. The management improves. The cleaning coverage improves. The crystal specification is maintained more efficiently every year. The fish eats. The civilization sees. Cross-references, Part 2, CH4, The Crystal Tube Standard, Part 4, CH3, Maintenance Without Humans, Part 4, CH6, Biological Civil Engineering, Part 5, CH2, Limestone Crustaceans, Part 5, CH6, Ecology as Infrastructure, Part 6, CH3, The Underground Safari, Part 8, Section B, The Living Pantry. For Cistrus vitreous acoustic management signal calibration protocol and optical transmission threshold deployment trigger, see Appendix F, Biological Operations Manual. For genetic drift monitoring loci and founding genotype archive maintenance schedule, see Appendix F, Biological Operations Manual. For food web productivity management through light relay photosynthetically active output control, see Appendix F, Biological Operations Manual. For network-wide reference standard revision protocol and annual biological management review format, see Appendix H, Governance Operations Manual. Pipe Dream. 5. The Engineered Ecosystem. Chapter 2, Limestone Crustaceans. The cave system the civilization inhabits did not produce its passages by waiting. It produced them by working, slowly, continuously, grain by grain, molecule by molecule, across geological time, through the accumulated action of water chemistry and biological processes on calcium carbonate at every scale from the molecular to the geological. The cenote network that Pipe Dream occupies is the product of 66 million years of this work. The work continues. The dissolution that produced the existing passages is still dissolving the passages' walls at the margin, widening some passages, deepening some chambers, occasionally producing the ceiling collapse events that the sacrificial caller's acoustic monitoring tracks as geological health indicators. The formation is not a static substrate the civilization inhabits. It is a dynamic geological system that is still making itself, on timescales that dwarf the civilization's design life but that intersected at specific points where the rate of geological change is fast enough to be relevant within a human lifespan. The boring program's lithocrustacean colony is the civilization's primary instrument for accelerating the formation's own production process, for expanding the passage network at a rate the civilization's design life can use rather than at the rate geological dissolution produces unaided. But the boring program is a construction instrument. It opens new passages. It does not maintain the passages it opens or the existing passages the crystal tube standard routes through. The boring program produces. The maintenance program sustains. The distinction is the same distinction that separates construction from civil engineering. The boring program is a builder, the maintenance program is the steward. The lithocrustacean boring colony is the builder. The limestone maintenance system is the steward. The stewardship of the formation's geological fabric, the management of the limestone that the crystal tube network inhabits, the management of the dissolution chemistry that is continuously working on that limestone, the management of the biological and chemical processes that are simultaneously stabilizing and destabilizing the passage geometry, is the geological stewardship function that the founding charter's water stewardship obligation addresses at the hydrological scale and that the limestone crustacean system addresses at the structural scale. The structural scale is where the civilization's thousand-year design life is most directly at geological risk. The anchor pylons connect the inhabited structure to the limestone. The limestone is dissolving. Not at a rate that matters over a decade. Not at a rate that matters over a century. At a rate that matters over 5 centuries, the design life's midpoint, and more acutely at a rate that matters in the zones where the installation's operations have altered the local dissolution chemistry, the zones where the ballast system's discharge chemistry has changed the water's mineral saturation state, where the biological communities the civilization manages have altered the organic acid production rate that drives limestone dissolution, where the anchor pylon's electrochemical potential in the formation water has introduced a galvanic component to the dissolution rate at the pylon-limestone interface. The civilization has altered the dissolution chemistry at its own foundations. The limestone crustacean system is the management response. What dissolves and why? Calcium carbonate dissolves in water containing carbonic acid. Carbonic acid is the product of carbon dioxide dissolving in water. Carbon dioxide enters the cenote's freshwater zone from 2 sources, the atmosphere, through gas exchange at the water surface in the cenote opening, and the biological community, through the respiration of every aerobic organism in the freshwater zone that consumes organic matter and releases carbon dioxide as metabolic waste. The inhabited cenote's biological community is denser than the uninhabited cenote's community, the food web the living pantry's aquaculture introduces, the ancestors vitreous colonies, the pink dolphin population, the biofilm communities on every installed surface, and produces more carbon dioxide per unit volume of freshwater zone than the unmanaged cenote's community produces. More carbon dioxide means more carbonic acid. More carbonic acid means higher dissolution rate at every limestone surface in contact with the freshwater zone's water. The increased dissolution rate is managed at the regional water quality level by the Water Stewardship Protocol. The coordinating systems monitoring confirms that the dissolution rate has not increased beyond the bounds the founding charter's pre-installation baseline allows. But the management at the regional scale does not address the local concentration at the anchor pylon-limestone interface, where the pylon's electrochemical potential in the cenote water produces a localized chemistry that the regional monitoring spatial resolution does not resolve. The pylon's electrochemical potential is a consequence of the stainless steel material in an electrolytic solution, the cenote water, which carries dissolved mineral ions at concentrations that make it electrically conductive. The stainless steel pylon is a galvanic element in this electrolytic environment. Its potential relative to the surrounding limestone produces a current flow in the pylon water limestone circuit that drives ion transport at the pylon limestone interface. The ion transport accelerates dissolution at the pylon limestone interface beyond the rate that the bulk water chemistry would produce in the absence of the electrochemical potential. The galvanic acceleration of dissolution at the pylon limestone interface is a material compatibility consequence that the anchor pylon specification addresses through material selection, 316L stainless steel in the freshwater zone, Inconel in the anoxic zone, both selected for minimum galvanic potential relative to the limestone in their respective depth zones water chemistry. Minimum galvanic potential is not zero galvanic potential. The dissolution acceleration exists even with optimal material selection. The limestone crustacean system manages the acceleration's consequence. The dissolution at the pylon-limestone interface is slower than it would be with a less compatible pylon material, but it is not zero, and across the thousand-year design life its accumulation is the structural concern the system addresses. The food forest soil's water chemistry adds a third source of dissolution chemistry alteration. The food forest irrigation, the ballast overflow and black water digestate that the agricultural management protocol delivers to the food forest's root zone, carries dissolved organic acids from the digestate's anaerobic decomposition products. These organic acids are plant nutrients at the concentrations the food forest's root zone receives them. They are dissolution accelerants at the concentrations that percolate through the cenote's limestone karst from the food forest's root zone toward the freshwater zone's water table. The concentration decreases dramatically during the percolation. The limestone sorption chemistry attenuates the organic acid concentration as the water passes through the pore network. By the time the food forest's irrigation water has percolated to the water table, the organic acid concentration is well within the range the bulk dissolution chemistry management maintains. But the percolation pathway, the limestone pore network between the food forest's root zone and the water table, experiences the full concentration gradient from the food forest's irrigation chemistry to the water table chemistry. The portions of this pathway closest to the root zone experience dissolution chemistry that the pore network's limestone is not in equilibrium with, producing localized dissolution along the percolation pathway. The localized dissolution along the food forest's irrigation percolation pathway is not a structural concern for the crystal tube installation. The percolation pathway is above the water table, in the vadose zone that no crystal tube section occupies. But it is a formation integrity concern for the food forest's cenote rim, the limestone between the food forest's root zone and the cenote opening. Dissolution along the percolation pathway widens the pore network. Wide enough pore networks, concentrated along specific flow paths by the dissolution chemistry's preferential attack on existing pore structures, can produce the conditions for subsurface collapse, the same mechanism that produces new cenote openings in the formation's geological natural history. The limestone crustacean system's geological stewardship function includes monitoring the vadose zone dissolution chemistry along the food forest's percolation pathways and managing the irrigation chemistry to maintain the dissolution rate within the range that the formation's limestone integrity assessment specifies as acceptable for the cenote rim's structural stability across the 1,000-year design life. The lithocrustacean stewardship variant. The boring colony lithocrustacean and the geological stewardship system's lithocrustacean are the same species with different deployment configurations. The boring colony's configuration, maximum population density at the bore face, boring appendage at maximum oscillation rate, paste output rate calibrated to the bore advance rate, is optimized for passage expansion. The stewardship configuration, lower population density distributed across the monitored surface area, boring appendage at minimum maintenance oscillation rate, paste output rate maximized relative to boring rate, is optimized for dissolution management and structural surface maintenance. The stewardship configuration operates in the maintenance mode that the coordinating system's biological management layer deploys rather than the boring colony's construction mode. The deployment zones are different. The boring colony advances through unexplored limestone ahead of the crystal tube network's front. The stewardship configuration occupies the zones behind the network's front, the passage walls and ceiling surfaces that the crystal tube installation has passed through and whose long-term structural integrity is the foundation the crystal tube anchors rest in. The stewardship configuration's boring appendage at minimum oscillation rate is not boring in the constructive sense. It is not producing bore advance. It is producing the acoustic microfracture characterization that the ground-penetrating sensor performs at the anchor sequence. The oscillation produces acoustic pulses that penetrate the surface limestone and return stratigraphic information about the subsurface limestone's dissolution state. The stewardship organism is a walking acoustic sensor, covering the passage wall surface at the movement rate the coordinating system specifies, returning continuous geological characterization data to the coordinating system's structural stability monitoring layer. When the acoustic characterization data identifies a dissolution zone, a region where the calcium carbonate's crystal structure has been weakened by dissolution to below the structural threshold the coordinating system's geological model maintains as the minimum for crystal tube anchor load support, the stewardship colony's response is the paste treatment. The boring appendage's oscillation increases to the fracture rate, the paste production is maximized, and the stewardship organisms at the identified zone concentrate their paste application at the dissolution weakened surface. The paste infiltrates the dissolution weakened crystal lattice through the microfractures the acoustic characterization oscillation has opened at the minimum fracture energy required to access the weakened zone, and the fast cure cementation reaction consolidates the dissolution weakened calcium carbonate back toward the structural threshold. The treatment is the same chemistry as the boring colony's tunnel sealing output, refined calcium carbonate paste, cemented to the substrate at the rock-water interface chemistry. The application is different. Instead of sealing a freshly fractured bore wall, the stewardship configuration is infiltrating and consolidating a dissolution weakened formation surface. The organism's biology is unchanged. The deployment context, the acoustic characterization mode replacing the bore advance mode, the paste production maximized instead of calibrated to bore advance rate, is the management configuration the coordinating system specifies. The stewardship lithocrustation cannot reverse dissolution. The calcium carbonate that has been dissolved into the formation water and carried away by the passage flow is gone from the formation and is not recoverable by the stewardship system. What the stewardship system can do is detect dissolution weakened zones before they reach the structural threshold below which crystal tube anchor loads cannot be safely supported, consolidate the dissolution weakened zone to above the structural threshold through paste infiltration, and reduce the dissolution rate at the treated zone by covering the freshly exposed calcium carbonate surfaces that the dissolution has produced with the paste's consolidated layer, reducing the reactive surface area that the dissolution chemistry can attack. The stewardship system is not winning against dissolution. Dissolution will continue, at the reduced rate the paste's coverage produces, in the consolidated zone, in the direction of the existing dissolution chemistry gradient. The stewardship system is managing dissolution to a rate and a spatial pattern that the crystal tube standard structural design life can accommodate, keeping the dissolution rate within the range that the structural monitoring's predicted remaining service life estimates project as compatible with the 1,000-year design life's anchor load requirements. The Anchor Zone Monitoring Program. The stewardship lithocrustaceans acoustic characterization of the anchor zone's limestone condition is the primary data source for the coordinating system's anchor integrity monitoring. The Anchor Pylon Inspection Protocol, the acoustic ranging survey that the census unit variant performs at each pylon at the inspection interval, provides the pylon material and grout interface data. The stewardship lithocrustaceans continuous acoustic characterization of the passage wall surfaces surrounding each anchor zone provides the geological context data, the dissolution state of the limestone that the anchor grout is bonded to. The two datasets combine in the coordinating system's structural health monitoring layer to produce the complete anchor integrity assessment, pylon condition, grout interface condition, and surrounding limestone condition. An anchor zone where all three indicators are within specification is an anchor zone the structural health monitoring categorizes as fully functional. An anchor zone where one indicator is approaching its threshold triggers increased monitoring frequency and the stewardship colony's targeted paste treatment of the approaching threshold element. An anchor zone where any indicator has crossed its threshold triggers the remediation protocol that Chapter 3 of this part documented for the anchor pylon's grout interface, injection grouting through a lithocrustacean board conduit, restoring the compromised element's structural specification. The Anchor Zone Monitoring Program's full dataset, pylon condition, grout interface condition, and surrounding limestone condition for every anchor pylon in the installation, is the coordinating system's structural health monitoring layer's primary product. The layer's output is a three-dimensional map of the installation's anchor integrity, which zones are fully functional, which are approaching thresholds, which are in active remediation, updated at the monitoring frequency each element's condition trend specifies. This map is the Terraform operator's primary structural intelligence briefing. The morning monitoring review that begins every Terraform operator's operational day includes the anchor integrity map's overnight update, the changes in the map since the previous day's review that indicate developing conditions requiring the Terraform operator's professional attention. A developing dissolution zone at an anchor pylon surrounding limestone is a condition the Terraform operator notes and monitors at increasing frequency as the zone approaches the stewardship colony's intervention threshold. A grout interface approaching threshold is a condition the Terraform operator notes and schedules for the injection grouting remediation before the threshold is crossed. The Terraform operator's anchor integrity monitoring is not passive review of sensor data. It is active geological stewardship, the professional responsibility for the limestone the civilization rests on, exercised daily through the monitoring review and exercised operationally through the concurrent authorization that the stewardship colony's targeted intervention and the injection grouting remediation each require. The Terraform operator does not authorize actions without data. The data does not produce actions without the Terraform operator's authorization. The partnership is constitutional, specified by the founding charter's governance architecture as the correct relationship between the coordinating system's analytical capability and the Terraform operator's professional judgment. Tunnel maintenance. The lithocrustacean boring program produces tunnels. The tunnels require maintenance across the 1,000-year design life. The maintenance is not the same operation as the boring. The boring produces the tunnel by advancing through solid limestone, and the maintenance manages the tunnel's stability after the boring has passed and the crystal tube installation has been completed. The tunnel's primary post-installation maintenance challenge is the paste lining's long-term integrity. The paste that the boring colony extruded as tunnel sealing output during the boring advance is calcium carbonate in a microstructural form that the cementation reaction has consolidated to a sealing layer. This sealing layer is not identical in mineralogy to the surrounding limestone. It has a different crystal structure, a different porosity, and a different dissolution rate than the natural limestone the boring program passed through. The natural limestone's dissolution rate, calibrated across millions of years of interaction with the cenote water chemistry, is in equilibrium with the water chemistry's current saturation state relative to calcium carbonate. The paste lining's crystal structure is not necessarily in the same equilibrium. The paste's precipitation chemistry was calibrated to the bore water's chemistry at the time of application, which may have changed in the years since application as the crystal tube installation's operations have altered the local water chemistry. The paste lining's long-term integrity assessment is performed by the stewardship lithocrustacean's acoustic characterization during its passage wall surveys. The paste lining's acoustic response to the stewardship organism's oscillation pulses differs from the natural limestone's acoustic response. The crystal structure difference produces a different acoustic impedance, and the coordinating system's acoustic monitoring layer is calibrated to detect both natural limestone and paste lining as distinct acoustic signatures. A paste lining in good condition, fully consolidated, no dissolution weakened zones, no microfractures that have propagated beyond the acceptable range, produces a characteristic acoustic response that the monitoring layer classifies as within specification. A paste lining approaching structural threshold produces a modified acoustic response that the monitoring layer flags for stewardship intervention. The stewardship intervention for a paste lining approaching structural threshold is a targeted paste reapplication. The stewardship organism positions its boring appendage at the flagged zone, oscillates at the minimum fracture energy to open microfractures in the compromised lining material, and directs the paste output into the opened microfractures. The reapplied paste consolidates the compromised lining back toward specification. The process is the same as the anchor zone's dissolution treatment, opening microfractures at minimum energy to access the compromised zone and filling those microfractures with fresh paste that cures to the structural threshold. The paste lining's long-term evolution is toward increasing similarity to the surrounding natural limestone, the diagenetic recrystallization that Chapter 5 of Part 4 described as the geological timescale integration of the boring program's passages into the formation's geological history. This evolution is not the stewardship system's enemy. The paste that is approaching geological maturity, approaching the crystal structure and dissolution equilibrium of the surrounding natural limestone, is paste that no longer requires stewardship treatment at the above natural rate. The stewardship treatment burden for each tunnel section decreases over geological time as the paste lining matures toward the natural limestone's equilibrium chemistry. The tunnels that require the highest stewardship intensity are the newest tunnels, where the paste lining's initial crystal structure is furthest from the natural limestone's equilibrium. The tunnels that require the lowest stewardship intensity are the oldest tunnels, where geological maturation has brought the paste lining toward the natural limestone's equilibrium. The stewardship burden curve across the network, high at the active boring front, decreasing through the established tunnel sections, approaching zero in the oldest sections, mirrors the construction burden curve. The newest work requires the most support, and the support requirement decreases as the work matures. The resource allocation that the coordinating system's stewardship scheduling produces tracks this curve. The stewardship colony's density and activity is highest near the active boring front, decreasing through the established network, and minimal in the oldest sections where geological maturation has made the formation's natural chemistry and the paste's chemistry indistinguishable. The cenote rim as geological structure. The cenote rim, the limestone above the water table that the cenote opening is bounded by, is the civilization's most geologically exposed structural element. Not in the engineering sense, the rim carries no crystal tube loads, no anchor pylon loads, no structural connection to the inhabited installation below. In the geological sense, the rim is the formation's most dynamic zone, receiving the full rainfall chemistry directly, experiencing the most aggressive dissolution chemistry from the rainfall's carbonic acid content before the water table's buffering chemistry attenuates the acidity, and carrying the food forest's irrigation chemistry through its vadose-pore network in direct concentration before the percolation path's limestone sorption chemistry reduces the organic acid load. The cenote rim's dissolution rate is higher than the submerged limestone's dissolution rate because the rainfall chemistry is more aggressive than the water table's buffered chemistry. Carbonic acid freshly produced from atmospheric carbon dioxide in rainwater is at higher concentration than the cenote water's carbonic acid, which has been in contact with the limestone's calcium carbonate for sufficient residence time to approach the dissolution equilibrium. The freshly formed carbonic acid in rainfall attacks the rim limestone with a greater driving force than the equilibrated cenote water attacks the submerged limestone. The cenote's natural geological history is a history of rim dissolution producing eventual rim collapse, the mechanism through which new cenote openings form. The cenote opening is the current expression of a rim collapse that occurred when the dissolution had weakened the rim sufficiently for the accumulated karst void below to roof fail. The current rim's dissolution is continuing the geological process that produced the opening, working on the rim that has not yet collapsed. Eventually, without civilizational management, the rim's dissolution would produce new structural weakness and eventually new collapse events. The stewardship system's geological monitoring of the vadose zone above the water table, the zone that conventional crystal tube monitoring does not reach, because no crystal tube section is installed in the vadose zone, is performed by a surface-deployed variant of the stewardship configuration. The surface-deployed variant operates in the food forest's root zone, accessing the vadose-pore network through the root channels that the food forest's deep-rooted species have opened through the limestone karst. The variant's boring appendage operates in the air-filled and partially air-filled pore network of the vadose zone rather than in the water-filled passages of the phreatic zone. The acoustic characterization oscillation in air-filled limestone pores produces different acoustic signatures than in water-filled limestone pores, and the coordinating system's geological model is calibrated for both signatures. The surface-deployed variance movement through the food forest root network is managed by the coordinating system's chemical gradient management, as with all lithocrustation deployment modes. The route guidance compound in this case is introduced through the food forest's irrigation system, dissolved at low concentration in the irrigation water, following the irrigation percolation pathway through the vadose zone, creating a concentration gradient that the surface-deployed variant follows from the food forest surface through the root channels into the vadose pore network. The stewardship organism follows the irrigation water's percolation path, the path through which the dissolution chemistry the stewardship system is managing is also flowing. The organism surveys the path that the chemistry is taking, characterizes the dissolution state of the limestone along that path, and reports the characterization data to the coordinating system through the acoustic transceiver network that the root channels support at low efficiency but sufficient bandwidth for the geological monitoring data's data rate. The surface-deployed variance paste application in the vadose zone is the geological equivalent of the phreatic zone's paste treatment, consolidating dissolution-weakened limestone surfaces with calcium carbonate paste that cures to the structural threshold. In the vadose zone, the paste application is targeted at the pore network's most dissolution-weakened elements, the pore throats whose constriction-controlling structures are closest to collapse, whose collapse would widen the pore network in a way that accelerates the percolation rate and the dissolution chemistry's exposure to fresh limestone surfaces. The management objective is not to halt vadose zone dissolution. Vadose zone dissolution has been proceeding since the limestone was deposited and will proceed until the formation itself is dissolved. The management objective is to maintain the vadose zone's pore network structural integrity at the scale of the cenote rim stability requirement, to prevent the dissolution from concentrating in the specific pore network configuration that would produce roof failure events within the 1,000-year design life's timeframe, in the specific locations where the food forest's root zone is proximate to the cenote rim. The food forest's roots and the stewardship organism's vadose zone survey and treatment occupy the same pore network channel. The roots stabilize the pore network through physical reinforcement. Root tissue in a pore channel provides mechanical resistance to the channel's collapse that the empty pore does not provide. The stewardship organism chemically consolidates the pore network through paste treatment. The root reinforcement and the chemical consolidation are complementary geological stewardship mechanisms, the biology of the food forest and the biology of the stewardship system working in the same pore network toward the same geological stability objective, each through its own mechanism, the combination more effective than either alone. The karst system as managed geology. The surface world manages geology in the sense of avoiding it, siting structures away from known geological hazards, designing foundations to transfer loads to competent geology below the surface geology's hazardous zone, and monitoring for geological events that threaten the structures built on and in geology. The surface world's geological management is passive and reactive: avoid the hazard, design around the hazard, monitor and respond to the hazard's expression. Pipedream manages the karst geology actively and prospectively, deploying biological systems that monitor the dissolution chemistry continuously, characterize the limestone structural state continuously, and apply consolidation treatment before the structural state crosses the threshold below which the crystal tube standards design assumptions are violated. The management is not after the fact. It is before the fact, intervention at the early dissolution stage, before the dissolution accumulation produces a structural event that the reactive response must address. This active geological management is only possible because the stewardship lithocrustation can operate at the spatial and temporal resolution that geological monitoring requires, continuously, at the passage wall surface scale, at the biological movement rate that covers the monitored surface area within the monitoring frequency the dissolution rate's progression speed requires. No human monitoring crew can operate continuously at passage wall surface scale. No mechanical sensor network provides the spatial resolution that a walking biological organism provides at the passage wall surface. The biological organism is the correct monitoring tool for the formation's geological condition because the biological organism's movement produces the spatial coverage that the geological conditions monitoring requires, at the temporal continuity that the dissolution rate's progression speed requires, in the chemical and physical conditions that human monitoring personnel and mechanical sensor alternatives cannot sustain in the formation's environments. The lithocrustation is doing geology because biology can do geology in the cenote's conditions, and because the geology the formation is doing to itself is the geology the civilization needs to understand and manage across the 1,000-year design life. The organism was designed to bore. The boring physics happened to produce the acoustic characterization that the geological monitoring requires. The organism was deployed to bore. The deployment to the monitoring zone, the established passage network rather than the boring front, produces the geological characterization without requiring a separate monitoring deployment. The boring instrument and the geological instrument are the same organism in different deployment configurations. One organism class, two functions, the second function emergent from the first function's physics applied in a different context. The formation's geology produced the cenote. The civilizational biology characterizes and manages the cenote's geology. The organism that connects the two, the lithocrustation, deployed at the boring front as the civilization's expansion instrument and deployed at the established network as the civilization's geological stewardship instrument, is the biological expression of the civilization's commitment to managing what it inhabits rather than extracting from it and leaving. The geological stewardship commons. The geological stewardship function, the monitoring, management, and consolidation of the limestone that the crystal tube standard rests in, is a commons function in the Redeemer governance framework. The limestone is not owned by the principality, the founding shareholders, or any individual cenote installation. The limestone is the formation, and the formation is the commons that the principality's sovereignty claim is built on and that the principality's water stewardship obligation extends to protect. The geological stewardship functions management, the stewardship colonies deployment protocols, the anchor zone Monitoring program standards, the vadose zone treatment protocols thresholds, the dissolution rate management's boundary conditions, is a Network Governance Council function under the REDEEM framework. No individual cenote installation's Terraform operator can revise the geological stewardship standards. The standards are network-level commitments that protect the commons resource, the limestone, that every installation in the network depends on. The governance rationale is straightforward. The dissolution chemistry that the installation's operations produce at one cenote's anchor zone affects the dissolution chemistry of the passage water that flows from that cenote through the inter-cenote connection passages to the adjacent cenote's freshwater zone. The karst is connected. The dissolution chemistry at one installation is not local, it is shared through the aquifer with every installation the connected passage network links to. The stewardship standard that one installation applies to its dissolution chemistry management affects every other installation's dissolution chemistry environment. This connectivity is the geological argument for network-level governance of the geological stewardship function. The commons resource is connected, and the management of one part of the commons affects every other part. The Network Governance Council's authority over the geological stewardship standards is the governance architecture's recognition of this geological connectivity, the management authority organized at the same scale as the resource it manages. The Terraform operators hold their concurrent authority at the installation level within the network standards the Network Governance Council establishes. The Terraform operator cannot exceed the network standards, cannot allow the dissolution rate at their installation to exceed the network-level boundary condition, but can apply the standards more conservatively if the specific installation's geological conditions warrant more conservative management. The network standard is the floor. The Terraform operator's judgment sets the ceiling above the floor at the level the specific installation's conditions require. The geological stewardship is the civilization's most fundamental commons management obligation, the management of the limestone the civilization is made of. The civilization cannot exist without the limestone. The limestone cannot sustain the civilization indefinitely without the geological stewardship that maintains the dissolution rate within the range the limestone's structural integrity across the 1,000-year design life requires. The governance that protects the stewardship function from the institutional pressures that would compromise it is the governance that protects the civilization from the geological consequence of the compromise. The REDEEM framework enforces this protection constitutionally because the geological consequence of insufficient stewardship is structural failure at geological timescales, the slow accumulation of dissolution damage to the anchor zones, the passage wall surfaces, the vadose zone's poor network, that compounds without observable expression for decades before the expression is a structural event that remediation cannot reverse. The protection must be constitutional because the timescale of the consequence is too long for the annual governance cycle to reliably sustain the protection through normal institutional processes. A governance council that can, within its normal authority, reduce the geological stewardship investment to fund a tourism infrastructure expansion will reduce the geological stewardship investment under sufficient economic pressure. The constitutional constraint prevents this by removing the reduction from the governance council's authority range. The limestone cannot speak at governance council meetings. The geological stewardship standard speaks for it. What the limestone crustacean knows. Like the Ancestris vitreus, the stewardship lithocrustacean knows nothing of what it is doing in the civil engineering sense. It knows the chemical gradient that guides its movement, the acoustic signal that indicates its boring appendage's target oscillation frequency, the paste production rate that the coordinating systems management specifies, the halt compound's priority response that withdraws it from the bore zone during seismic risk events. It does not know it is monitoring geological dissolution. It does not know it is consolidating anchor zone limestone. It does not know it is managing the vadose zone's dissolution chemistry above the water table that the civilization depends on. It is doing the biology the founding biologists specified, moving, oscillating, extruding, responding to chemical and acoustic signals, and the civil engineering consequence of this biology, in the specific deployment context the coordinating system manages, is geological stewardship. The stewardship is in the deployment context. The biology is in the organism. The two together produce the geological management the civilization requires. This is not a surprising observation at this point in the document. The same observation applies to the Ancestris vitreus cleaning the tube walls, to the mycofiltration panel's fungal network processing the gray water, to the food forest's mycorrhizal network distributing soil nutrients. All of these organisms are doing their biology. The civil engineering function is in the context the civilization has provided for the biology. What is worth noting specifically about the lithocrustacean in this context is the scale of what the organism's biology is managing. The civilization rests on limestone. The limestone is dissolving. The dissolution rate determines the civilization's structural foundation's durability across the 1,000-year design life. A designed organism that produces acoustic characterization data as a byproduct of its boring oscillation and consolidation paste as a byproduct of its feeding metabolism is managing the dissolution of the civilization's structural foundation with its own sensory output and metabolic waste products. The civilization's foundation is being managed by the biological byproducts of an organism the size of a hand. The organism does not know it is supporting the civilization. The civilization knows the organism is supporting it. The founding biologists specified the organism to support it. The coordinating system deploys the organism to support it. The terraform operators authorize the deployment. The Network Governance Council sets the standards. The organism crawls across the limestone, oscillating, extruding, responding to signals, eating the material the oscillation produces, returning the eaten material as paste to the wall it crawled across. The limestone is a little more consolidated where it has been. The dissolution rate is a little slower at the surface it has coated. The anchor pylons surrounding limestone is a little more capable of supporting the structural loads the crystal tube imposes. The passage wall is a little more stable than it would be if the organism had not been there. Multiply this by thousands of organisms, over decades, across the full surface area of the passage network, and the geological consequence is the difference between the formation that the Structural Monitoring's 1,000-year design life assessment projects as structurally adequate and the formation that the same assessment would project as approaching inadequacy without the stewardship program's intervention. The organisms are not saving the civilization. The civilization does not need saving by organisms the size of a hand. The organisms are maintaining the geological foundation at the rate the Biological Management Protocol specifies, the rate the geological monitoring data indicates is required, the rate the Structural Monitoring's 1,000-year projection requires to remain above the structural threshold. The organism crawls. The limestone consolidates. The civilization stands. The limestone crustacean and the living underworld's longevity. Part 3 established the living underworld as the foundation on which parts 5 through 12 are built. The living underworld rests on the limestone. The limestone's geological integrity is the living underworld structural prerequisite. The lithocrustacean's geological stewardship function is the maintenance of that prerequisite across the 1,000-year design life. No other chapter in Part 5 describes a system more foundational to the civilization's physical existence than the lithocrustacean's geological stewardship. The cleaner fish maintains the crystal tube's optical function. The geological stewardship maintains the crystal tube's structural foundation. The optical function matters because the crystal specification is the civilization's visual contract with the formation. The structural foundation matters because without it the crystal tube does not stand and the crystal specification has no medium to be maintained in. The hierarchy is, formation geology supports structural foundation supports crystal tube standard maintains crystal specification enables visual transparency enables the civilization's contract with the formation. The lithocrustacean at the geological stewardship tier maintains the first link in this chain. Every other system in the installation chain depends on this first link holding. The founding charter established the geological stewardship obligation as a constitutional commitment because the founding engineers understood this dependency. The Biological Management Protocol specifies the stewardship colony's deployment standards because the founding biologists understood the organism's capability to address the obligation. The terraform operators exercise their concurrent authority in the anchor zone monitoring review because the governance architecture understood that the obligation must be exercised by a professional with the biological and geological training to assess whether the stewardship standard is being met. The limestone crustacean is not a spectacular organism. It does not produce the visual drama of the pink dolphin's passage through the illuminated freshwater zone, or the biological spectacle of the living pantry's aquaculture galleries, or the medical significance of the longevity program's telomere results. It crawls on limestone walls in the dark, oscillating, extruding paste. But the civilization is built on the limestone it crawls on. The paste it extrudes is the geological glue that holds the limestone together under the load the civilization imposes. The acoustic characterization its oscillation produces is the geological intelligence the coordinating system uses to decide where the geological glue needs to be applied next. The civilization's longevity is 1,000 years. The limestone crustacean's geological stewardship makes it possible. Cross-references, Part 1, CH4, Floating Before Anchoring, Part 2, CH6, Designing for 1,000 Years, Part 3, CH1, Rivers Beneath the Jungle, Part 4, CH3, Maintenance Without Humans, Part 4, CH5, The Living Tunnel Makers, Part 4, CH6, Biological Civil Engineering, Part 5, CH1, Cleaner Fish, Part 9, CH2, Floating Tidal and the Habitation Record. For stewardship configuration deployment protocol and acoustic characterization oscillation frequency calibration by lithological zone, see Appendix F, Biological Operations Manual. For anchor zone monitoring program standards and dissolution rate boundary condition specification, see Appendix A, Formation Baseline Protocol. For Vado zone monitoring protocol and food forest irrigation chemistry management parameters, see Appendix F, Biological Operations Manual. For Network Governance Council Geological Stewardship Standards and Terraform Operator Concurrent Authority Boundaries, see Appendix H, Governance Operations Manual. Pipe Dream. Part 5, The Engineered Ecosystem. Chapter 3, Pink Dolphins. The boto is not a mythological creature. The surface world's Amazonian folklore has made it one, the encantado, the shapeshifter, the river spirit that assumes human form to seduce and be seduced, Because the boto's intelligence, its navigational capability in turbid water, its apparent curiosity about human activity at the river's surface, and its striking pink coloration in mature individuals all combine to produce an animal that the human pattern recognition system wants to classify as something more than an animal. The boto resists classification as ordinary wildlife. It does not behave like ordinary wildlife. It navigates through flooded forests in the Amazon basin's wet season by echolocation, tracking fish through root networks and submerged vegetation in conditions of zero visibility, exhibiting what field researchers have consistently described as something that looks, at insufficient caution's distance, like problem solving. The boto's intelligence is not human intelligence. The boto's navigation is not rational planning in the human sense. The boto's apparent curiosity about human activity is not the social interest of a species with a theory of mind comparable to the primate families. But the boto's cognitive architecture, the acoustic processing that converts echolocation returns into spatial models of complex three-dimensional environments, the social memory that maintains individual recognition across years of separation, the behavioral flexibility that adapts hunting strategies to prey behavior changes in real time, is sophisticated beyond what most vertebrate nervous systems achieve, and its sophistication is exactly calibrated to the navigation challenges that in Amazon floodplain submerged forest environments present. The Yucatan cenote cave system is not the Amazon floodplain submerged forest. The geological substrate is limestone rather than tropical hardwood root networks. The water chemistry is freshwater aquifer rather than floodplain inundation. The biological community is the karst cave fauna's ecological successor community rather than the Amazon basin's hyperdiverse tropical freshwater community. The navigation challenges are similar in one critical respect: complex three-dimensional environments with limited visibility, requiring acoustic spatial modeling at the resolution that prey tracking demands. The founding biologist's identification of the boto, Aenia geofrensis, as the biological chassis for the cenote's apex freshwater predator was the identification of a cognitive architecture pre-adapted to the navigation challenges the cenote presents. Not through modification but through recognition, the boto's evolved navigation capability is the navigation capability the cenote's spatial complexity requires, and the cenote's spatial complexity is not significantly different, from the acoustic navigation perspective, from the Amazon floodplain's submerged forest complexity. The animal that evolved to navigate one is already equipped to navigate the other. The modification was the cenote chemistry's adaptation requirements, not the navigation architecture. The navigation architecture was already correct. The bioengineered variant. Aenia geofrensis in its natural form inhabits the Amazon and Orinoco river systems' freshwater environments, rivers, floodplains, and connected lakes across a geographic range that includes multiple distinct population groups with divergent evolutionary histories. The natural boto is adapted to warm freshwater at low altitude, to high turbidity conditions that make visual navigation irrelevant, and to the Amazon basin's specific water chemistry, the Black Water River's acid, low conductivity, tannin-stained chemistry, and the White Water River's sediment-loaded, higher conductivity chemistry produce different osmoregulatory demands that Amazon boto populations have adapted to regionally. The Yucatan cenote's freshwater chemistry is calcium bicarbonate type, alkaline, clear, with dissolved mineral concentrations substantially higher than the Amazon's Black Water Rivers and lower than the White Water Rivers. The natural boto's osmoregulatory biology, tuned to the Amazon's chemistry ranges, requires modification to maintain the ion regulation efficiency the cenote's different mineral ion profile demands. The modification target was the kidney tubules ion transport proteins, the specific molecular components whose substrate affinity determines which ion concentrations the kidney can regulate against, and across what concentration ranges the regulation remains energetically sustainable. The kidney modification was the primary physiological change. Secondary modifications addressed the cenote's different light environment. The cenote's clear water transmits light from the cenote opening to depths that the Amazon's turbid water never illuminates, creating visual input that the natural boto's light-limited sensory system is not fully equipped to use. The cenote-adapted variant's visual cortex allocation was increased relative to the natural boto's, providing greater visual processing capacity for the cenote's illuminated zones while retaining the full echolocation architecture that the cave passage's unilluminated sections require. The unfused cervical vertebrae that the master context brief identifies as a navigation asset, the anatomical feature that allows the cenote-adapted boto to execute 90-degree head turns, is not a modification. It is a preserved ancestral feature that the natural boto's family group retained when the rest of the cetacean lineage fused the cervical vertebrae for hydrodynamic efficiency in open-water high-speed swimming. The boto's ecological niche in the Amazon basin never demanded the open-water acceleration that rewarded vertebral fusion in oceanic dolphins. The floodplain's obstacle-rich navigation rewarded head mobility. The unfused vertebrae persisted as the adaptation for a navigation environment that obstacle density makes more important than hydrodynamic efficiency. The cenote's cave passages are more obstacle-dense than the Amazon floodplain. The cenote-adapted boto's cervical vertebral mobility is the anatomical feature most directly relevant to its passage navigation capability. A boto that can turn its head 90 degrees while maintaining forward body orientation can execute acoustic characterization of passage geometry that a rigid-necked swimmer cannot achieve, can direct the echolocation beam toward the passage wall, receive the return, and redirect toward the passage ceiling or the passage floor in a continuous 360-degree spatial characterization sweep without requiring the body to turn. The 90-degree head turn is the difference between the passage being a tunnel the boto passes through and being a space the boto maps in 3 dimensions while transiting it. The head turn's ecological significance for the dolphin survey function, documented in Chapter 2 of Part 3 as a primary source of passage geometry data for the coordinating system's aquifer layer, is a preserved ancestral feature applied to a novel ecological context. The founding biologists did not engineer the unfused cervical vertebrae into the cenote-adapted variant. They selected the boto as the chassis specifically because the boto's evolutionary history had preserved this feature that the cenote's navigation demands require. The biology was already there. The deployment context made its engineering value visible. Echolocation in karst. The boto's echolocation is not a simple distance ranging system. It is a high-resolution acoustic imaging system that constructs spatial representations of complex three-dimensional environments from the amplitude, frequency, and temporal structure of returning acoustic echoes. The natural boto navigating a submerged forest produces clicking trains at rates and with directional beam patterns calibrated to the prey tracking problem, high-rate clicks at narrow beam angles for precise target ranging, low-rate clicks at wide beam angles for environmental mapping. The navigation behavior switches between these modes as the task changes: environmental mapping during transit, prey tracking during hunting, social acoustic exchange during social interaction. The cenote's karst limestone presents acoustic environments the Amazon basin's submerged forest does not. Limestone is denser than water and transmits acoustic energy at higher velocity than water. The acoustic impedance difference between water and limestone is larger than the impedance difference between water and submerged wood. The boto's echolocation click that reaches a limestone passage wall and reflects does not simply echo back from the wall surface, it transmits partially into the limestone, reflects from internal structures within the limestone, and returns to the boto as a complex multipath signal carrying information about the wall's surface geometry, the wall's interior geology, and the passage geometry on the other side of the wall if the wall is thin enough for the transmitted component to return a detectable reflection. The cenote-adapted boto's acoustic processing system was modified to extract the limestone's multipath reflection signature's geological information. Not to replace the prey tracking and environmental navigation functions that the natural boto's acoustic processing performs, these are retained and remain primary, but to add a geological characterization processing layer that interprets the wall-transmitted reflection components as geological information rather than noise. The natural boto's acoustic processing discards the multipath reflection components as noise. The cenote-adapted boto's modified processing layer classifies them as signal, as geological data about the wall's interior and the passage geometry beyond. This is not a trivial modification. The geological characterization processing layer requires the acoustic system to maintain the full environmental navigation and prey tracking function at the same time as it processes the multipath reflections for geological information, parallel processing of the same acoustic input stream for 2 different analytical purposes simultaneously. The neural substrate for this parallel processing was the modification's most technically demanding component, the acoustic cortex's reorganization to support parallel processing without degrading the navigation function's performance. The result is an animal whose echolocation serves 3 simultaneous functions: navigation in complex 3-dimensional limestone environments, prey tracking at the spatial resolution that the cenote's fish prey require, and geological characterization of the limestone passage walls at the resolution that the coordinating system's aquifer layer can use as passage geometry input. The 3 functions use the same clicking behavior, the same returning acoustic stream, and the same acoustic processing neural substrate, organized in parallel rather than sequentially, producing 3 outputs from 1 sensory process. The coordinating system's dolphin survey extraction, the processing of the biointegrated transponder tags recorded click return pairs into passage geometry estimates, accesses the geological characterization output of the cenote-adapted boto's acoustic processing. The dolphin maps the limestone. The coordinating system reads the map. The dolphin does not know it is mapping the limestone for the coordinating system. It is echolocating for navigation and prey tracking in the way that its biology, natural and modified, produces. The geological characterization is the processing layer's output from the same acoustic input the navigation and prey tracking layers process. The dolphin echolocates. The coordinating system intercepts the geological characterization layer's output. The aquifer map grows. The halocline as biological wall. The halocline that the crystal tube standards engineering must cross, the zone whose optical dissolution effect defines the most distinctive visual experience in the installation, whose temperature gradient drives the thermal management challenges documented in Chapter 3 of Part 2, whose electrochemical potential drives the halocline salinity gradient battery's power generation, is, for the cenote-adapted boto, an absolute boundary. The boto cannot cross it. The boundary is not behavioral. It is physiological. The halocline salinity gradient imposes an osmoregulatory demand that the cenote-adapted Boto's kidney modification cannot address across the full salinity range between the freshwater zone and the saltwater zone. The modification tuned the ion transport proteins for the cenote's freshwater calcium bicarbonate chemistry. It did not extend the ion transport capacity into the saltwater regime. The Boto at the halocline boundary experiences the same osmotic stress that any freshwater cetacean would experience at a marine salinity interface, the water drawing fluid across the skin and gill membranes faster than the kidney can compensate, producing dehydration at the cellular level and the neurological consequences that cellular dehydration produces. Death at the halocline is not a rapid event. It is the accumulation of ionic imbalance across the time the animal spends in the halocline zone. The Boto knows the halocline before it reaches the halocline. The echolocation detects the acoustic impedance difference between the freshwater column and the saltwater column below the halocline boundary. The density difference between the two water masses produces an acoustic reflection that the Boto's echolocation processes as a structural boundary. The Boto experiences the halocline acoustically as a surface below the freshwater column, analogous to the bottom boundary of a river pool. The natural Boto's behavior at a pool's bottom boundary is to avoid contact with the boundary while exploiting the prey concentration that the boundary's upwelling creates. The cenote-adapted Boto's behavior at the halocline is the same behavioral pattern applied to the acoustic signature of the halocline: approach the boundary for prey exploitation, but not cross it. The coordinating system's light array behavioral deterrent, the specific pulse frequencies at the halocline boundary depth that serve as a gentle behavioral reinforcement of the Boto's natural halocline avoidance, is a management supplement to a behavior that the Boto's physiology and echolocation already produce. The deterrent prevents the specific behavioral states that might override the Boto's natural halocline avoidance, the excited prey pursuit that drives a Boto following a fast fish toward the halocline at high speed, where the momentum and the prey fixation might carry the animal across the acoustic boundary before the osmoregulatory distress signals registered. The light pulse at the halocline boundary is a sensory interrupt, a visual stimulus in the frequency range that the cenote-adapted Boto's expanded visual cortex is sensitive to, that triggers behavioral reorientation before the osmoregulatory threshold is reached. The deterrent is not coercive. The Boto's sensory system responds to the light pulse as it responds to any sudden stimulus in its visual field, with the orienting reflex, the reflexive reorientation of attention toward the stimulus source, interrupting the current behavioral sequence to assess the stimulus. The orienting reflex breaks the prey pursuit chain at the halocline boundary. The Boto reorients, reassesses, and the prey that crossed the halocline boundary, which the coordinating system's prey management protocol allows to cross the halocline because the prey species can survive the crossing, is no longer the Boto's pursuit target. The Boto returns to the freshwater zone's prey pool. The halocline deterrent system is not preventing the Boto from doing something it wants to do. It is interrupting a behavioral chain, excited prey pursuit, before the chain's momentum produces a physiological consequence, halocline crossing, that the Boto's biology would not choose if the physiological consequence were the decision criterion rather than the prey stimulus. The deterrent gives the Boto's biology a moment to be the decision criterion instead of the prey stimulus. The freshwater zone's acoustic ecology. The Boto is not the freshwater zone's only acoustic actor. The Ancestris vitreous colonies produce low-frequency bioluminescent coordination signals that the freshwater zone's water column transmits at attenuated amplitude. The maintenance ultrasonic operations, the squeegee bot fleet's operations in the halocline zone, below the Boto's habitation zone but within the acoustic propagation range of the freshwater zone's water column, produce tonal output that the Boto's acoustic system detects as background. The scavenger communications that adventure guests use in the freshwater zone produce acoustic energy at the communications frequency range. The crystal tube network's maglev propulsion system produces electromagnetic flux that the Boto's lateral line detects as current variations. The acoustic ecology of the freshwater zone is not the acoustic ecology that the cenote's pre-installation biological community experienced. The installation has added acoustic sources that the cave fauna's evolutionary history has not produced adaptation responses to. The cave fish species that inhabit the freshwater zone alongside the Boto, the blind, depigmented, lateral line-dependent species that the cenote's pre-installation biological census documented, are experiencing acoustic conditions their evolutionary history did not prepare their sensory systems to filter. The Boto's acoustic processing system handles the added acoustic sources through the parallel processing architecture that its modification produced, classifying the maintenance frequency sounds as background noise, the scavenger communications as non-prey acoustic events, the maglev flux as non-acoustic environmental variation. The cavefish's simpler sensory systems do not have the same filtering capacity. The coordinating system's acoustic zoning protocol, documented in Chapter 2 of Part 2 as the management system that prevents the installation's acoustic outputs from interfering with the coordinating system's instruction channel and the biological management signals, also manages the installation's acoustic impact on the freshwater zone's biological community. The zoning protocol is not primarily a welfare protocol for the cave fish. It is an engineering protocol for the communication channel's integrity. But the cavefish's acoustic exposure is a parameter the biological management protocol tracks through the biological census's behavioral indicators, stress response frequencies in the cavefish population, changes in spatial distribution that indicate acoustic avoidance behavior, changes in reproductive activity that reflect elevated chronic stress levels. The Bodo's acoustic management is the acoustic zoning protocol's most demanding scheduling constraint. The Bodo's social vocalizations, the high-frequency burst pulse sounds and the low-frequency groans that Bodo populations use for mother-calf communication, inter-individual recognition, and social coordination, are acoustic events that the acoustic zoning protocol cannot suppress or redirect. The Bodo's vocalizations are the Bodo's biology. The protocol does not manage the Bodo's vocalizations. It manages everything else around them, scheduling the maintenance ultrasonic operations, the scavenger communications, and the squeegee bot fleet's acoustic output in windows that the Bodo population's vocalization activity is at its minimum, as characterized by the biological monitoring network's continuous passive acoustic recording of the Bodo population's activity patterns. The Bodo's vocalization pattern is therefore the acoustic zoning protocol scheduling input, the fixed acoustic presence that the protocol arranges all other acoustic activities around. The Bodo does not accommodate the installation's acoustic schedule. The installation accommodates the Bodo's acoustic biology. This accommodation has a practical consequence for the adventure program's scavenger excursion scheduling. The scavenger excursions that take guests into the freshwater zone for direct encounter with the Bodo population are scheduled in the vocalization minimum windows that the Bodo's activity pattern creates, early morning before the social activity that the daylight column's arrival stimulates, and late afternoon after the hunting activity that the midday hours produce has concluded. The scheduling serves the acoustic zoning protocol's communication channel integrity, the cavefish community's acoustic stress management, and the guest experience's quality simultaneously. The Bodo populations encountered during vocalization minimum windows are resting and socially calm populations whose approach tolerance for scavenger-equipped guests is higher than the tolerance of actively hunting or socially engaged populations. 1 scheduling parameter, 3 simultaneous outcomes. The formation's biology sets the parameter. The installation accommodates it. The accommodation serves multiple functions. Population genetics in the closed system. The cenote-adapted Bodo population faces a genetic challenge that the natural Bodo does not face in its Amazon basin habitat. The cenote's freshwater zone is a closed volume. The natural Bodo's Amazon habitat, despite the Amazon basin's complex geography, maintains connectivity between population groups through seasonal flood events, river channel migrations, and the connected river systems that link the basin's disparate water bodies. Gene flow between natural Bodo populations, while limited by the distances involved, occurs at sufficient frequency to maintain genetic diversity within each population and prevent the inbreeding depression that genetically isolated populations accumulate across generations. The cenote's freshwater zone has no seasonal flood connectivity to other water bodies. The halicline prevents connectivity with the aquifer saltwater zones. The crystal tube network's inter-cenote connections are the only connectivity paths available for gene flow between installed cenote's Bodo populations, and the connectivity paths are managed by the behavioral deterrent arrays and the terraform operator's joint authorization requirement for inter-cenote transits documented in Chapter 3 of Part 3. The population genetics management challenge is therefore a managed connectivity challenge, providing sufficient gene flow between cenote populations to prevent inbreeding depression accumulation while managing the connectivity through the crystal tube network's transit infrastructure rather than through the unmanaged connectivity of natural habitat connectivity. The metapopulation management framework that the regional network establishes, each cenote installation hosting a local population with managed genetic exchange coordinated by the Network Governance Council's biological management standards, is the governance response to the geological isolation that the cenote's freshwater zone imposes on its Bodo population. The effective population size management is the metapopulation framework's primary genetic management challenge. Effective population size, the number of individuals that contribute equally to the next generation's gene pool, adjusted for variance in reproductive success, sex ratio imbalance, and population size fluctuations, determines the rate of genetic drift in the population. Genetic drift is the random change in gene frequencies that occurs in finite populations regardless of selection pressure, the random sampling error in reproduction that, over generations, produces allele frequency changes that have nothing to do with the allele's adaptive value. In small populations, genetic drift is rapid. In large populations, genetic drift is slow. The cenote Bodo population's effective population size is constrained by the freshwater zone's carrying capacity, the prey resource base that the freshwater zone's biological productivity sustains at the prey density that supports the Bodo population's nutritional requirements. A freshwater zone that sustains a dozen Bodo individuals has an effective population size that produces genetic drift at a rate incompatible with genetic integrity across the 1,000-year design life. 12 individuals reproducing through genetic drift for 1,000 years produce a population whose genetic diversity is a small fraction of the founding population's diversity, and whose inbreeding depression has reduced the population's biological fitness to levels that the biological management protocol cannot address through habitat management alone. The minimum viable population size for genetic integrity across the 1,000-year design life, the minimum number of breeding individuals whose effective population size produces a genetic drift rate that the managed intersynoty gene flow can offset, was calculated by the founding biologists before the first Bodo was introduced to the founding cenotes freshwater zone. The calculation determined the minimum freshwater zone volume, the minimum prey resource base, and the minimum managed gene flow frequency from adjacent cenote populations that a viable installation Bodo population requires. Every cenote installation's freshwater zone was specified to the minimum volume and prey resource base that the viable population size calculation requires. The aquaculture management protocol in the freshwater zone maintains the prey resource base at the level that the viable population requires. The managed gene flow between cenote populations, the intersynote Bodo transits that the terraform operators schedule, authorize, and execute through the crystal tube network's corridor connection, is specified at the frequency the effective population size calculation requires to offset the isolated population's drift rate. The frequency is not high, even a few individuals per generation exchanged between cenote populations is sufficient to maintain genetic diversity at a level that prevents inbreeding depression within the management framework's design life parameter. The transit frequency requirement is typically 1 to 3 individuals per generation per connected cenote pair, depending on the effective population sizes of both populations and the genetic distance between them at the managed loci. The Biological Monitoring Network's genetic census, tissue samples collected from Bodo individuals during the veterinary examination protocol that the Autonomous Veterinary Robotics Program conducts, sequenced at the critical genetic loci, and compared against the founding population's genetic composition and the network-wide genetic diversity database, tracks the managed gene flow's adequacy. If the genetic census identifies an individual cenote's population drifting away from the network-wide diversity baseline faster than the current intersynote transit frequency can offset, the transit frequency recommendation for that cenote pair is increased. If the census confirms that the current frequency is maintaining the population's genetic diversity within the management target's range, the frequency is maintained at its current specification. The genetic management is continuous, data-driven, and population-specific. Not a fixed transit schedule applied uniformly across the network. A dynamically adjusted schedule calibrated to each population's current genetic state relative to the network-wide target. The coordinating system generates the schedule. The terraform operators authorize the transits. The Bodo individuals complete the transits. The genetic diversity is maintained. Behavioral ecology in the managed habitat. The cenote-adapted Bodo in the managed freshwater zone is not a wild animal in a controlled environment. It is an animal whose behavioral ecology has developed in the specific conditions of the managed freshwater zone, conditions that share much with the natural Bodo's Amazon habitat and differ from it in specific ways that the cenote's geology, the installation's infrastructure, and the management protocol's interventions produce. The similarities are the behavioral foundations that the natural Bodo's evolutionary history produced, the echolocation-based navigation, the prey pursuit and capture strategies, the social structure organized around female kinship groups with male competition for mating access at the periphery, the extended juvenile dependency that the Bodo's cognitive development requires. These behaviors are present in the cenote-adapted Bodo because they are the behavioral architecture of the Bodo chassis the founding biologists began with. The management protocol did not produce them. It maintains the conditions under which they can express. The differences are the behavioral adaptations the cenote-adapted Bodo has developed across the generations since the founding installation's first Bodo introduction. The founding individuals encountered the crystal tube network, the light relay illumination, the acoustic management signals, the scavenger-equipped guests, the ancistrous vitreous schools on the tube walls, the aquaculture gallery spectra of fish species in the living pantry corridor sections, all as novel stimuli without behavioral history in the species' evolutionary or individual experience. The founding individuals' responses to these novel stimuli were exploratory and variable. Some individuals approached the crystal tube walls and echolocated at them intensively, apparently interested in the acoustic multipath returns from the tube material. Some individuals avoided the light relay's illuminated sections until hunger drove them into the illuminated zone's prey population. Some individuals engaged with scavenger-equipped guests with the curiosity that Bodo individuals consistently display toward novel objects in their environment. The behavioral responses the founding individuals expressed toward the installation's novel elements have been transmitted to subsequent generations through the social learning that Bodo populations use for behavioral transmission in the Amazon basin's prey-catching techniques and navigation routes. The cenote-adapted Bodo's fifth-generation individuals were born in a freshwater zone whose crystal tube network, light relay illumination, acoustic management signals, and occasional scavenger-equipped guests were not novel stimuli. They are the environment. The behavioral responses to these environmental features that the founding individuals developed through exploratory engagement have been transmitted through social learning to subsequent generations as established behavioral patterns rather than as exploratory responses. The most consequential behavioral transmission is the Bodo population's relationship to the aquaculture gallery sections that the living pantry corridor cenote connections introduce to the crystal tube transit routes. The founding individuals' response to the aquaculture galleries, densely stocked fish schools in transparent mesh cylinders, illuminated by the light relay, at close approach distance from the transit corridor, was intense predatory interest. The founding individuals learned, within their first season of access to the aquaculture gallery sections, that the mesh cylinder enclosures prevented direct prey access. They did not learn to stop attempting prey access. The predatory motivation is not extinguishable by experience of enclosure. They learned to exploit the prey that was available at the aquaculture gallery sections, the spillage from the harvest drone operations, the juvenile fish that escaped through mesh apertures smaller than the harvest drone's awareness threshold, the invertebrate prey that the aquaculture infrastructure's surface area supports. This exploitation of the aquaculture gallery section's spill and escape population is an unplanned benefit of the living pantry corridor cenote's biological management. The Bodo population in the connected freshwater zone harvests the aquaculture gallery's unintentional prey surplus at no cost to the aquaculture production system's intentional output. The Bodo does not reduce the aquaculture production. It harvests what the production system does not intend to produce and would otherwise re-enter the broader freshwater zone food web through the biological community's trophic structure. The Bodo is the aquaculture gallery's largest bodied scavenger, collecting the spill and escape population that the aquaculture management protocol's harvest accounting does not track. The behavioral transmission of the aquaculture gallery section's exploitation strategy to subsequent generations has produced a foraging route preference in the cenote-adapted Bodo population that concentrates the population's activity near the crystal tube network's living pantry corridor sections at the harvest drone's active hours. The population's spatial distribution within the freshwater zone is not random. It is structured by the behavioral transmission of prey opportunity knowledge, exactly as the natural Bodo spatial distribution in the Amazon basin is structured by the behavioral transmission of prey opportunity knowledge across generations of social learning. The managed habitat has produced managed behavior. The management was not designed to produce this specific behavioral outcome. The behavior emerged from the interaction of the Bodo's cognitive architecture, the aquaculture gallery's prey surplus, and the social learning that Bodo populations have always used to transmit prey opportunity knowledge. The management provided the conditions. The biology produced the behavior. The BOTO as apex indicator. The freshwater zone's ecological health is readable through the Bodo population's demographic indicators in a way it is not readable through any other single biological parameter. The Bodo is the freshwater zone's apex predator, the highest trophic level in the food web that the freshwater zone's biological community supports. Apex predators in food webs integrate the ecological health of all the trophic levels below them. Their population size, body condition, reproductive rate, and behavioral health reflect the cumulative condition of the prey populations they depend on, the water quality conditions that affect their physiology, and the habitat conditions that support their behavioral ecology. A Bodo population in declining condition in a food web where all lower trophic levels are healthy indicates a condition-specific impact at the apex predator's specific biology, a pathogen affecting the Bodo but not its prey species, a toxin accumulated at the apex trophic level through bioaccumulation from low concentration sources in the prey population, or a habitat condition change that affects the Bodo's specific ecological requirements without affecting the prey species whose ecological requirements are different. A Bodo population in declining condition in a food web where the lower trophic levels are also declining indicates a systemic food web impact, water quality degradation, prey population reduction, or habitat disruption that cascades through the food web from the lower trophic levels upward. The Coordinating Systems Biological Monitoring Protocol uses the Bodo population's demographic indicators as the freshwater zone's primary ecological health index. Because the apex predator's integrated response to the food web's cumulative condition provides diagnostic information about the food web state that the coordinating system's lower trophic level monitoring supplements but cannot replace. The Bodo's body condition, measured by the veterinary examination protocol's morphometric assessment during each individual's examination, reflects the prey population's nutritional quality. The Bodo's reproductive rate, measured by the genetic census's reproductive success analysis, reflects the habitat condition's adequacy for successful reproduction. The Bodo's social structure stability, assessed through the Behavioral Monitoring Network's group composition tracking, reflects the population's absence of significant chronic stress. These three indicators together form a comprehensive freshwater zone ecological health assessment that no combination of water chemistry measurements, fish population counts, and biofilm community surveys alone provides. The Bodo's biology integrates the food web's cumulative state into its own demographic condition. Reading the Bodo's condition is reading the food web's health through the lens of its apex consumer. The Terraform Operator's Biological Management Review includes the Bodo population's current demographic indicators alongside the water chemistry monitoring data, the fish population census, and the Ancestris vitreous colony's crystal specification compliance rate. The Bodo's indicators are not interpreted in isolation from the other data. They are interpreted in the context of the other data, the Bodo's condition explained by the food web's current state, the food web's current state explained partly by the water chemistry explained partly by the ballast systems management. The ecological interpretation chain connects the Boto's body condition to the ballast management protocol through the food web's trophic structure. The Boto speaks for the freshwater zone. The freshwater zone speaks for the installation's water management. The water management speaks for the civilization's operational discipline. If the Boto is not well, the installation asks why. The answer traverses the ecological chain from the apex predator's body condition to the mechanism that produced the condition. The mechanism's identification is the corrective action's prerequisite. The Boto's condition is the signal. The ecological chain is the diagnostic path. The terraform operator is the diagnostician. The Boto's relationship to the coordinating system. The relationship between the Boto population and the coordinating system is the most complex organism management relationship in the installation. The Ancestris vitreous is directed by acoustic management signals to specific cleaning locations. The lithocrustation is guided by chemical gradient signals along specific boring routes. The pink dolphin is managed through the acoustic deterrent at the halocline boundary, the light pulse deterrent at the halocline boundary, the veterinary examination protocol, the intersynody transit scheduling, and the prey resource-based management through the food web's productivity control. None of these management mechanisms command the Boto's behavior. The deterrents interrupt behavioral chains that would produce physiologically harmful outcomes. The prey resource management shapes the food web that the Boto's foraging behavior responds to. The veterinary examination provides health data that the biological management protocol uses to optimize the habitat conditions. The intersynody transit scheduling provides managed connectivity that the Boto's own movement through the crystal tube corridors executes. The Boto is not a managed animal in the sense that the Ancestris vitreous is a managed animal, directed to specific locations at specific times to perform specific functions. The Boto is an animal living in a managed habitat, a habitat whose conditions are managed to support the Boto's behavioral ecology while producing the ecological functions the installation requires of the freshwater zone's apex predator. The difference is significant. The managed animal's behavior is the management's product. The Boto's behavior is the Boto's product, expressed in a habitat whose management makes specific behaviors more likely, less likely, or impossible. The Boto chooses, in the non-deliberative sense that an animal's behavioral architecture choices operate, how to navigate its habitat. The coordinating system manages what the habitat offers for the Boto to navigate. This distinction is the founding biologist's most important governance insight about the synoty-adapted Boto. An animal that is directed to produce specific behaviors will produce those behaviors for as long as the direction produces them and will not produce them when the direction fails. An animal that is living in a habitat whose conditions support its natural behavioral architecture will produce the installation-relevant behaviors, the geological survey echolocation, the aquaculture spill harvest, the halocline avoidance, the social vocalization patterns that the acoustic zoning protocol accommodates, as long as the habitat conditions support the behavioral architecture. The habitat management is more resilient than the behavioral direction. The habitat is a system, the direction is a signal. Systems survive component failures. Signals stop when the source fails. The coordinating system can fail. The habitat can fail. The Boto's behavioral architecture cannot fail in the same sense. It is the product of millions of years of evolutionary selection for navigating exactly the kind of environment the cenote presents. The management that maintains the habitat conditions is more durable than the management that directs the behavior, because the habitat is what the behavior is already designed to navigate. The Boto is not the installation's employee. It is the freshwater zone's resident. The freshwater zone is the installation. The Boto lives in what the installation is. The installation is designed to be a place where the Boto's behavioral architecture produces what the installation's ecological function requires. This is the cleanest expression of aquaforming at the biological tier, not designing the organism's behavior to produce the required function, but designing the habitat so that the organism's natural behavioral architecture produces the required function in the natural course of living there. What the Boto produces. The Boto produces prey population regulation that prevents any single prey species from dominating the freshwater zone's fish community to the exclusion of the species diversity the ecological management protocol maintains. Geological survey data through the echolocation returns that the coordinating system's dolphin survey extraction processes. Acoustic mapping of passage geometry ahead of the boring program's transceiver chain reach through the echolocation's three-dimensional passage. Characterization, behavioral data about the freshwater zone's food web condition through the demographic indicators the biological monitoring protocol tracks, and the visual and acoustic presence that makes the freshwater zone's inhabited volume legible to guests as a living ecosystem rather than as a flooded cave. The last output is not trivial. The freshwater zone's visual character, the illuminated water column, the biological community on the crystal tube walls and the natural limestone surfaces, the food web's trophic dynamics visible through the transparent tube walls, would be ecologically coherent to a biologist but not immediately legible as a living system to a guest without prior exposure to cave biology. The Boto's presence makes the freshwater zone immediately legible as a living system to any observer regardless of biological training. An organism the size of a large dog, pink, visibly alive, visibly purposeful in its movement, echolocating with clicks the guest can hear through the tube wall, navigating the passage geometry with apparent ease. This is the freshwater zone's biological reality made unmistakably visible to the unaided human perception. The Boto is the freshwater zone's ambassador to the human observer's perception. Not in the way that an animal exhibit's ambassador species serves the exhibit's educational function, curated for visual impact, positioned for observation, performing for the audience. The Boto is the freshwater zone's ambassador because the Boto is the freshwater zone's apex organism, and apex organisms make ecosystems legible at the apex observer scale. The human observer perceives the freshwater zone as a coherent living system because the freshwater zone has an apex organism whose behavior represents the system at a scale the human perception can engage with. The ecosystem is real. The Boto is real. The legibility is the Boto being what it is in a habitat that the installation has made real. The ambassador is not performing. The ambassador is living. The living is the performance. The BOTO and the civilizational thesis. The founding charter's description of pipe dreams found in cognitive advantage, the sans a priori, the absence of inherited orthodoxy that allows every design decision to be made on first principles calibrated to what the formation actually offers, is nowhere more completely expressed than in the decision to introduce a large marine mammal into the cenote's freshwater zone as primary infrastructure. The surface world's instinct, faced with the challenge of managing a complex flooded cave network, would be to manage the biology out, to filter the water, sterilize the surfaces, exclude the fauna, and install the infrastructure in a biologically neutralized environment whose biological management challenges are reduced to zero by the elimination of the biology. The surface world's infrastructure paradigm treats biology as a management problem whose optimal solution is the biology's removal. The founding engineers and biologists faced the same cenote and concluded the opposite: the biology is the infrastructure. The cave's biological community is the maintenance system, the monitoring system, the survey system, and the ecological resilience the installation's long-term stability requires. The biology is not the problem. The biology is the solution. The problem is designing the infrastructure to work with the biology rather than against it. The Boto is the fullest expression of this conclusion. An apex predator in the freshwater zone was not a management challenge the founding team chose to accept. It was a design decision the founding team chose to make, the decision that an installed freshwater zone without an apex predator is an ecologically incomplete zone whose food web stability is lower, whose prey population regulation is absent, whose ecological legibility is reduced, and whose geological survey capability is zero. The Boto's presence addresses all four deficits simultaneously. The Boto's management challenges, the acoustic ecology management, the halocline deterrent, the population genetics program, the intercynody transit coordination, the veterinary protocol, are the cost of having an apex predator. The cost is worth paying because the alternative, an ecologically incomplete freshwater zone managed more simply but less effectively, is a worse installation. Sans a priori made this decision possible. The surface world's instinct to manage biology out, the inherited orthodoxy that biology and infrastructure is a complication, not an asset, would have produced a simpler management challenge and a worse outcome. The absence of that orthodoxy produced the Boto. The Boto swims in the cenote because the founding generation asked what the cenote could sustain before deciding what to put in it, and the cenote's freshwater zone can sustain an apex freshwater cetacean with unfused cervical vertebrae and a modified acoustic processing system that maps the cave's limestone passages while hunting fish. The cenote sustains it. The Boto lives there. The civilization benefits from both. The pink dolphin is pink. It is real. It is in the cenote. It is infrastructure. Cross-references, Part 3, CH2, Mapping the Great Aquifer, Part 3, CH3, Linking cenote to cenote, Part 3, CH4, Underground Boulevards, Part 3, CH6, The First Regional Network, Part 4, CH6, Biological Civil Engineering, Part 5, CH6, Ecology as Infrastructure, Part 6, CH3, The Underground Safari, Part 8, Section B, Pink Dolphins, Partners, Not Performers, Part 11, CH2, The Nitrox Galleries. For cenote-adapted Boto founding genotype specification and kidney modification protocol, see Appendix F, Biological Operations Manual. For metapopulation management framework, intercynody transit frequency calculation, and genetic census protocol, see Appendix F, Biological Operations Manual. For halocline deterrent light array specification and acoustic zoning protocol integration, see Appendix F, Biological Operations Manual. For dolphin survey extraction methodology and aquifer layer integration protocol, see Appendix G, Formation Intelligence Record. For autonomous veterinary examination protocol, see Part 8, Section B and Appendix F, Biological Operations Manual. Pipe dream. Part 5, The Engineered Ecosystem. Chapter 4, Aquaculture Cities. The surface world's aquaculture is a two-dimensional industry. The fish pond is a horizontal surface, water contained in a basin, with the fish population distributed across the basin's area and managed from the basin's perimeter. The cage net in an open bay is a three-dimensional volume in principle, but managed as a two-dimensional surface in practice. The feeding happens at the top, the waste accumulates at the bottom, and the management interventions, feeding, monitoring, harvest, are surface operations that access the volume's interior only for specific interventions. The two-dimensional management paradigm persists even in three-dimensional structures because the management infrastructure, the boats, the feeding systems, the monitoring equipment, operates at the water surface, not inside the volume. Surface world aquaculture scales by adding surface area, more ponds, more cages, more bay leases, more coastal real estate. The resource constraint is horizontal. The growth direction is horizontal. The three-dimensional volume of water below the surface is largely unmanageable and therefore largely unused by the aquaculture system, present, containing water and waste and disease organisms, but not producing food proportional to its volume. The cenote offers no surface to expand across. The cenote's planned dimension, the circular opening in the limestone canopy, is fixed by the dissolution geology that produced it. A 40-meter cenote opening is a 40-meter cenote opening. No expansion of the aquaculture system can make it 60 meters. The horizontal dimension is bounded by the limestone the dissolution process produced, and the dissolution process has not enlarged further on any timeline the civilization's planning horizon can use. What the cenote offers that the surface world's aquaculture has never systematically exploited is depth. The freshwater zone descends 15 meters from the cenote opening's water surface to the halocline boundary. The cenote's circular cross-section, maintained across this depth range, produces a cylindrical volume whose capacity is not the circle's area but the circle's area multiplied by the depth. A 40-meter diameter cenote with 15 meters of freshwater zone depth contains over 18,000 cubic meters of freshwater volume, the equivalent, in water volume, of several large surface fish ponds stacked vertically in a cylinder the size of a municipal swimming pool's planned footprint. The aquaculture city is the architecture of food production in that vertical cylinder. The architecture of vertical production. The cenote's vertical cylinder is not uniform as a food production environment. The vertical axis that Part 3 and Part V's preceding chapters have established as the civilization's ecological gradient, the light field, the temperature, the dissolved oxygen, the biological community composition all changing with depth, is also the aquaculture system's production parameter gradient. Different species require different light intensities, different temperatures, different dissolved oxygen concentrations, different prey and nutritional inputs. The species that a well-designed aquaculture city produces are not all at the same depth. They are stratified by depth, each species occupying the depth zone whose conditions match its biological requirements. This stratification is not a management challenge to overcome. It is the aquaculture city's organizational principle, the feature that allows a single cenote's water volume to support multiple species simultaneously without the interspecies competition that an unstratified mixed species system would produce. Each species occupies its zone. The zones are vertically stacked. The total production is the sum of the zone's production. The cenote's volume is used, not wasted in the vertical dimension. The uppermost production zone, the surface tier and the first few meters below the water surface, in the full illumination of the daylight column and the light relay, is the photosynthetic production zone. Spirulina bioreactors, vertical glass tubes of 100 to 200 millimeters diameter, sealed at both ends, with a peristaltic pump circulating the culture medium from the bottom upward. The culture medium is cenote freshwater enriched with the mineral nutrient concentrations the spirulina's growth rate requires, nitrogen, phosphorus, potassium, iron, zinc, manganese, in the proportions that the coordinating system's growth rate monitoring has determined optimizes the biomass production rate in the founding cenote's specific water chemistry. The light relay's photosynthetically active output is the bioreactor column's energy source. The column's exterior surface is exposed to the light relay's projection. The spirulina culture inside receives the light through the glass wall and converts it to biomass at the photosynthetic efficiency that the culture density and the light intensity together determine. The bioreactor column is not a passive cylinder. It is a continuously monitored production unit. Dissolved oxygen sensors at the column's midpoint and top register the photosynthetic rate through the oxygen produced. Temperature sensors register the thermal load the light relay imposes on the culture medium. Optical density sensors register the biomass concentration that determines the culture's current growth phase and the harvest timing. The coordinating system's aquaculture management layer processes all three sensor streams for each bioreactor column and adjusts the peristaltic pump rate, controlling the culture medium's vertical mixing and the light exposure time per unit of culture, to maintain the column's growth rate at the optimum that the current light intensity and culture density produce. The spirulina harvest is a partial harvest. The coordinating system identifies the harvest timing from the optical density sensor's biomass concentration curve, which reaches a peak that subsequent culture density increase would push past the self-shading threshold, the point at which the culture is dense enough that the interior cells receive insufficient light for optimal growth rate, reducing the column's volumetric productivity. The partial harvest removes enough biomass to return the column to the growth phase density below the self-shading threshold. The harvest cycle is continuous, not batch. Some fraction of the column's biomass is harvested every few days rather than the full column being cleared and restarted periodically. The continuous partial harvest maintains the column in its optimal growth phase indefinitely. The harvested spirulina biomass serves three simultaneous functions in the cenote's integrated production system: direct human food production for the installation's residents and guests, feed supplement for the fish species in the aquaculture city's deeper zones whose nutritional profiles benefit from the spirulina's protein and pigment content, and soil amendment material for the food forest's mycorrhizal network alongside the mycofiltration panel harvests. 3 outputs from one production unit, each output serving a different functional layer of the installation's integrated metabolism. The mid-column fish zone. Below the photosynthetic production zone, from 3 to 10 meters below the water surface, in the light relays transmitted illumination at lower intensity than the surface zone but still sufficient for the visual hunting behavior of the fish species the zone supports, the aquaculture city's primary protein production is organized in cylindrical mesh cages suspended from the surface tier's structural framework. The cylindrical mesh cage is the aquaculture city's primary production unit in the mid-column zone. Not because it is the only possible structure for fish aquaculture, but because the cylinder is the geometry that the cenote's round cross-section most naturally accommodates, that the coordinating system's current monitoring infrastructure can most efficiently address with sensor arrays deployed from the cage's central axis, and that the siltvac ROV fleet can most efficiently maintain through access from the cylinder's open bottom end. The cylinder's interior volume is the fish's living space. The mesh wall is the physical boundary between the production volume and the cenote's ambient water, fine enough to prevent fish escape and coarse enough to allow water exchange. Water exchange is the cylinder's waste management mechanism. The cenote's ambient water flow, induced by the crystal tube network's ballast pumping and the underground river's passage through the cenote's connected fracture network, moves through the mesh wall continuously, removing dissolved metabolic waste and introducing oxygenated water from the ambient column. The cylinder is not a sealed tank requiring mechanical water circulation. It is a permeable volume that the cenote's own water movement maintains within the biological water quality parameters the fish species requires. The species assigned to each depth position in the mid-column zone are selected by the coordinating system's aquaculture planning layer from the production portfolio species inventory on the basis of 3 simultaneous criteria. The species' biological requirements match the target depth zone's conditions. The species' production cycle timing fits the harvest schedule that the installation's food system demand forecasting specifies, and the species' trophic interactions with the other species present in the mid-column zone are managed within the spatial separation the cage geometry provides. The trophic separation management is the mid-column zone's most demanding design parameter. Several of the fish species in the aquaculture city's production portfolio are predatory. They will consume the smaller fish species if the spatial separation between cages is insufficient to prevent encounter. The cylinder geometry surface-to-volume ratio and the mesh wall's visual transparency determine the visual encounter rate between predatory and prey species in adjacent cages. The coordinating system's aquaculture planning layer models the encounter rate as a function of the cage spacing and the species' visual range in the ambient light conditions at the target depth, and specifies the cage spacing that produces an encounter rate below the behavioral stress threshold that impairs the prey species' production rate. The cage spacing specification produces the mid-column zone's horizontal layout, the arrangement of cages in the cenote's cross-section at each depth tier. Not all depth tiers accommodate the same number of cages. The deepest mid-column tiers have smaller cenote cross-sections in cenotes whose walls converge with depth, reducing the number of cages that the trophic separation specification allows. The aquaculture city's horizontal layout at each depth tier is the geometric output of the trophic separation specification applied to the cenote's cross-sectional dimensions at that depth. The cenote's geology determines the production capacity. The species selection determines the separation requirement. The separation requirement and the cenote geometry together determine the production unit count. The giant freshwater prawn, a decapod crustacean of the family Macrobrachidae, bioengineered for the cenote's calcium bicarbonate water chemistry, occupies the mid-column zone's upper depth band, where the water temperature is warmest in the freshwater zone and the current velocity from the crystal tube network's ballast flow provides the oxygenation the prawn's high metabolic rate requires. The prawn's benthic feeding behavior in the cage environment, grazing the cage wall's algal and biofilm accumulation, scavenging the organic material that settles from the surface production zone above, converts what would otherwise be waste material in the mid-column zone into prawn biomass. The prawn is the mid-column zone's detritivore, the trophic role that converts the upper zone's production residuals into harvested protein. The freshwater lobster, a parastacid crayfish variant adapted for the cenote's chemistry, occupies the mid-column zone's lower depth band, where the temperature drop toward the halocline and the reduced light intensity from the light relay's transmitted beam at this depth produce conditions the freshwater lobster's cold-adapted metabolism performs best in. The freshwater lobster's territorial behavior in natural habitat is managed in the cage environment through the cage geometry's vertical structuring, artificial reef materials, textured polymer panels with apertures sized for individual lobster occupancy, installed within the cylinder provide territorial space for each individual, reducing the agonistic interaction that unstructured close-density captivity in a territorial species produces. The freshwater lobster's territorial behavior management through structural habitat enrichment is not a welfare intervention applied secondarily to a production unit designed for maximum density. It is a production optimization. Territorial animals in unstressed conditions grow faster, convert feed more efficiently, and display lower mortality than territorial animals in chronic agonistic stress. The artificial reef materials cost is recovered in the production rate improvement they produce. The welfare outcome and the production optimization are the same outcome from the same intervention. The deep column zone and the halocline interface. The deepest freshwater zone aquaculture, the production at the bottom of the 15-meter freshwater column, within 2 to 3 meters of the halocline boundary, operates in conditions that few surface world aquaculture systems approach and none inhabit permanently, ambient pressure of approximately 1.1 to 1.15 atmospheres, temperature at the lower end of the freshwater zone's gradient, light from the relay at low intensity, and the chemical gradient of the halocline's salinity and H2S. Beginning to register in the dissolved chemistry measurements at sensor resolution even though the halocline's main boundary is still meters below. The production species at the deep column zone are selected for their tolerance of these marginal conditions and their exploitation of the biological productivity that the halocline boundary concentrates. The halocline is not a biological desert. It is a biological transition zone. The boundary between two water masses with different chemistries creates concentration gradients in dissolved nutrients, dissolved gases, and biological material that support elevated biological productivity relative to either mass alone. The halocline's chemocline, the zone of elevated chemical gradient that the two water masses contact produces, supports a microorganism community whose primary productivity is higher than the ambient freshwater community's productivity in the water column above it. This halocline productivity provides a nutrition source that the deep column zone's filter feeders exploit. The engineered mussel variants, bivalves modified for the cenote's freshwater chemistry and the halocline zone's atypical pressure and light conditions, are suspended in the deep column zone on vertical dropper lines extending from the cage framework above. The mussel's filter feeding extracts phytoplankton and particulate organic matter from the water column passing through its ciliary feeding apparatus. At the deep column zone's depth, the water column's particulate organic matter concentration is elevated by the halocline chemocline's productivity. The mussel is positioned at the depth where its food source is most concentrated. The engineered mussel's modification from its marine bivalve ancestor is primarily in the osmoregulatory biology. The freshwater cenote's calcium bicarbonate chemistry requires different ion transport management than marine salinity environments. The modification was the same kidney and gill ion transport modification that the Bodo cenote adaptation required, applied to the bivalve's mantle epithelium rather than the cetacean's kidney tubule. The feeding behavior, the shell deposition biology, and the reproductive cycle were retained from the ancestral chassis without modification. The marine bivalve's filter feeding is already the correct biology for the halocline zone's particulate food source, and the reproductive biology is compatible with management at the cenote's temperature and pressure conditions with only the scheduling adjustment the coordinating system's aquaculture planning provides. The mussel's filter feeding is also the deep column zone's water quality management function. The mussel clears particulate organic matter from the water column that, if allowed to accumulate, would increase the dissolved organic carbon concentration in the deep freshwater zone to levels that accelerate biofilm growth rates on the crystal tube surfaces at this depth and degrade the water chemistry quality that the ballast circuit's deep intake specifies. The mussel produces food while simultaneously maintaining the water quality the production zone's other biological systems require. The water quality management and the food production are the same organism doing its feeding biology. The Siltvac ROV fleet manages the deep column zone sediment accumulation, the organic waste from the deep column zone's mussel filtration, the fish metabolic waste from the cages above, and the particulate settling from the surface production zone that has passed through the mid-column zone's cages without biological uptake. The Siltvac ROV is a neutrally buoyant disc-shaped vehicle whose downward-facing suction system collects the sediment accumulation from the cage bottoms and the deep column zone's floor. The collected material is directed through a suction line to the deep anoxic zone below the halocline, where the anaerobic bacterial community processes it through the same metabolic cycles that the chemostat manages at depth. The organic waste from the freshwater zone's aquaculture city becomes the anoxic zone's feedstock, and the anoxic zone's metabolic products return as mineral harvest and methane power generation in the cycle that Part 10 documents. The Siltvac is the aquaculture city's waste management system. The anoxic zone is the waste management system's processing plant. The chemostat is the processing plant's intelligence layer. 3 different parts of the civilization's vertical extent, connected by the Siltvac suction line through the halocline boundary, cycling the aquaculture city's production waste back into the civilization's industrial metabolism. The loop has no exit. The waste is feedstock. The feedstock becomes product. The product returns to the freshwater zone as mineral inputs to the light relay's photosynthetically active outputs productivity enhancement. The genetics of production. Every species in the aquaculture city's production portfolio carries a genetic specification in the Coordinating Systems Biological Management Archive. The specification records the founding individual's genetic composition at the loci that determine the production-relevant characteristics: growth rate, feed conversion efficiency, disease resistance, reproductive timing, behavioral responses to confinement conditions, tolerance of the cenote-specific water chemistry parameters. The specification is the production baseline, the genetic configuration that produces the performance the aquaculture planning layer's harvest schedule is based on. Genetic drift in the production species' captive populations is managed by the same principle that governs the Bodo population's genetic management: periodic genetic exchange with a master genetic archive that holds the founding specification in cryogenic preservation, introduced to the production population when the genetic monitoring identifies drift from the specification at production-relevant loci. The genetic exchange for production species is simpler than for the Bodo metapopulation. The production species' smaller body size allows higher individual counts per genetic exchange introduction, and the shorter generation time of most production species produces more generations per monitoring period, giving the drift monitoring more data resolution per year of monitoring than the Bodo's longer generation time provides. The production portfolio's most commercially important genetic parameter is disease resistance. Aquaculture production systems globally have a documented relationship between high-density production and elevated disease incidence. The density-dependent stress response reduces individual immune function, and the high local concentration of host tissue creates the transmission opportunities that opportunistic pathogens exploit. The cenote aquaculture city's density is controlled by the cage spacing specification's production density limit, a density calibrated to maintain the population below the threshold at which density-dependent immune suppression produces disease susceptibility above the background pathogen load in the cenote's freshwater zone. The background pathogen load in the cenote's freshwater zone is monitored by the Coordinating Systems Water Quality Monitoring Network at the same sensor infrastructure as the water chemistry monitoring. Pathogen indicator organisms measured by the same sampling protocol as the dissolved chemistry parameters, at the same sampling frequency, with the results integrated into the aquaculture management layer's production density decision. If the pathogen indicator organisms' concentrations increase, a signal that the background pathogen load is elevated above the baseline for reasons that may or may not be related to the aquaculture operations, the production density limit is reduced proactively, below the standard density-dependent immune suppression threshold, to increase the population's immunological reserve against the elevated pathogen load. The disease management is conservative because the consequence of disease outbreak in the aquaculture city is not a single cage's loss, it is the potential for cascade through the cenote's connected freshwater zone if the pathogen is transmissible between cages and between species. The cage mesh walls that allow water exchange also allow pathogen transmission through the water column. A disease outbreak that progresses to a multi-cage, multi-species cascade is a production system disruption that the Biological Management Protocol's response framework is designed to prevent, not to manage after the fact. Prevention requires the conservative density management that allows the population's immunological reserve to contain the background pathogen load before any pathogen reaches the threshold for clinical outbreak expression. The genetic specification's disease resistance loci are the population's first immunological defense. The density management is the second. The water quality management that the mussels filter feeding and the siltvacs waste removal provide is the third. 3 layers of disease management, each operating through different mechanisms, each providing a margin of protection that the other 2 layers supplement. No single layer is sufficient alone. The combination is sufficient for the production density the aquaculture planning layer specifies. Harvest logistics. The aquaculture city's production is not batch harvested. The production species' staggered cage introduction, coordinated growth monitoring, and individual cage harvest scheduling produces a continuous harvest flow rather than the periodic mass harvest events that surface world aquaculture batch production generates. The continuous harvest flow matches the installation's food system's continuous demand. The longevity program's guests and the permanent residents require food every day, not once per production batch cycle. The harvest drone is the aquaculture city's primary harvest unit. The harvest drone is a neutrally buoyant vehicle approximately 30 centimeters in diameter, operating on the Crystal Tube Network's guide rail infrastructure in the passages adjacent to the aquaculture galleries, moving between the guide rail and the cage interior through the mesh apertures that the cage specification includes for drone access at the cage bottom. The drone's harvest mechanism is a selective suction system. The drone approaches a fish or prawn or lobster individual identified by its optical sensor as meeting the harvest size criterion, calibrated by species to the minimum size that the production schedule specifies for harvest, activates the suction briefly, collects the individual in the drone's internal holding chamber, and moves to the next harvest candidate. The harvest selectivity is the drone's primary value over the batch net harvest methods that surface world aquaculture uses. The batch net method collects all individuals in the cage simultaneously, requiring sorting post-harvest to separate harvest size individuals from sub-harvest size individuals. The sub-harvest size individuals return to the water from the sorting process experience handling stress. The capture, sorting, and return sequence imposes physiological stress that reduces growth rate in the returned individuals for several days post-handling. In a production system where growth rate optimization is the primary production efficiency parameter, several days of stress-reduced growth across the sub-harvest portion of the population represents a significant production efficiency cost. The drone's selective harvest avoids this. Only harvest size individuals are removed from the cage. Sub-harvest size individuals are never contacted by the harvest drone unless the optical sensor's size assessment produces a classification error, which the coordinating system's harvest accuracy monitoring tracks as an error rate metric and uses to calibrate the optical sensor's size threshold specification. The classification error rate target is low enough that the stress handling cost of classification errors is below the threshold at which the selection benefit is negated. The drone's harvest does not disturb the uncollected population. The uncollected population's growth rate is unimpacted by the harvest event. The harvest drone's collected individuals are transferred at the cage access point to the collection sub, a larger neutrally buoyant vehicle that follows the harvest drone's collection route along the guide rail, receiving the drone's filled holding chambers and transferring them to the collection sub's larger holding capacity. The collection sub's trajectory through the aquaculture city follows the species sequence that the harvest schedule specifies: spirulina partial harvest from the bioreactor columns, prawn collection from the mid-column upper zone, fish species collection from the mid-column central zones, lobster collection from the mid-column lower zone, mussel collection from the deep column zones dropper lines. The full harvest collection is completed in a single collection sub transit through the aquaculture city's vertical extent. The collection sub transfers its harvest to the processing gallery, a dedicated gallery in the cenote hub's working level, not in the tourist transit zones, where the harvest is sorted, processed, and packaged for the installation's food service and for the intercenote transit system's food distribution logistics. The processing gallery is the aquaculture city's downstream connection to the installation's food system. The aquaculture city produces protein at depth. The processing gallery converts it to food at the hub level. The food service distributes it through the installation's culinary infrastructure. Part 8's Food as Tourism chapter documents the full distribution chain's experiential design. The aquaculture city's contribution to that chain begins with the harvest drone's selective collection and ends at the processing gallery's transfer point. The aquaculture city as water treatment system. The aquaculture city's food production function is its explicit design purpose. Its water treatment function is the designed in consequence of the biological management that the food production system requires. The two functions are inseparable. They use the same organisms, the same infrastructure, and the same management operations to produce simultaneously different outputs: food from the aquaculture city's biology, and water quality from the aquaculture city's metabolic activity. The mussel's filter feeding at the deep column zone is the most direct expression of this inseparability. The mussel clears the water column of particulate organic matter as a byproduct of feeding on the particulate organic matter it has cleared. The clearing is both the feeding mechanism and the water treatment output. The food production and the water treatment are the same biological process observed from two different functional perspectives. The prawn's detritivore feeding at the mid-column zone's upper depth band is a second expression. The prawn consumes the organic detritus settling from the surface production zone, converting material that would otherwise accumulate as dissolved organic carbon in the water column into prawn biomass. The dissolved organic carbon's accumulation would degrade the water quality of the deep freshwater zone in ways that would eventually impair the mussel's filtration efficiency and the deep column zone's crystal tube wall's crystal specification compliance. The prawn prevents this accumulation by consuming the detritus at the source depth before it can accumulate at the depth where its accumulation would matter. The fish species metabolic waste, the ammonia and dissolved nitrogen that fish excretion adds to the water column, is the water quality parameter that surface world intensive aquaculture most commonly fails to manage within the bounds that the fish species can tolerate. In a surface pond or cage, ammonia accumulates faster than the water exchange can dilute it to acceptable concentrations at high production densities. The cenote aquaculture city's water quality management for metabolic ammonia depends on the same mechanism that manages it in natural aquatic systems: biological nitrification. The biofilm communities on the cage exterior mesh walls, the biological community that the Ancestris vitreous schools manage at the crystal tube walls, is also present on the cage walls. At the fouling density, the cage mesh's surface chemistry and the mid-column zone's water chemistry support include the nitrifying bacteria that convert ammonia to nitrite and nitrate. The nitrate at low concentrations is a nutrient for the photosynthetic community in the surface production zone above. The biofilm community on the cage wall surfaces is simultaneously a fouling management challenge for the cage's structural integrity. Accumulated biofilm on the mesh wire increases the mesh's hydrodynamic resistance to water flow, reducing the exchange efficiency, and the aquaculture city's ammonia management system. The coordinating system's cage management protocol maintains the cage wall biofilm at a density that optimizes the balance between nitrification rate and hydrodynamic resistance, enough biofilm to maintain effective nitrification, not so much that the water exchange efficiency is degraded below the dissolved oxygen supply requirement for the cage's fish population. The coordinating system achieves this balance through periodic partial biofilm removal by Ancestris vitreous deployment to the cage exterior mesh surfaces, the same acoustic management deployment that directs the cleaning school to the crystal tube walls, reprogrammed for the cage wall's mesh geometry. The Ancestris vitreous's enzymatic cleaning at the cage wall removes the outer biofilm layers that have exceeded the nitrification optimizing density while leaving the base layer that the coordinating system's water quality monitoring confirms is performing the nitrification function at the required rate. The cage wall biofilm is managed for dual function, nitrification performance and mesh hydrodynamics, through the same cleaning biology that manages the crystal tube wall for optical performance. The cleaning biology's application domain is extended from the crystal tube wall to the cage wall by acoustic management signal redirection. The organism does not change. The surface it cleans changes. The acoustic management configures which surface is cleaned, when, at what frequency. The biology is generic. The management is specific. Species selection and the formation's constraints. The aquaculture city's production species portfolio was not assembled from the commercially most valuable species available to freshwater aquaculture globally. It was assembled from the species that the cenote's specific chemical, thermal, pressure, and light conditions can support, filtered by commercial value, filtered by compatibility with the cenote's managed biological community, filtered by the lithocrustacean colony's boring program's ability to produce the freshwater zone volumes the production portfolio requires at the densities the harvest schedule specifies. The cenote's freshwater calcium bicarbonate chemistry excludes the acid-tolerant species that many tropical freshwater aquaculture systems exploit, the species whose commercial value in global markets is high but whose biological requirements include the acidic, low-conductivity water chemistry that the cenote's mineral-rich aquifer does not provide. The cenote's freshwater chemistry is alkaline and mineral-rich, conditions that favor hard water-tolerant freshwater species whose commercial value in global markets is significant but whose cultivation in surface world systems typically requires the water treatment infrastructure to maintain the alkaline, high-conductivity chemistry that occurs in the cenote naturally. The cenote produces the chemistry that hardwater freshwater aquaculture requires as a design target. Pipe Dream does not need to manufacture the water chemistry. The formation provides it. The production portfolio is built around species whose biological requirements match what the formation provides rather than species whose biological requirements require the formation's chemistry to be altered. This is aquaforming applied to the production species portfolio. The production portfolio conforms to the formation's chemistry rather than the formation's chemistry being engineered to conform to the production portfolio's requirements. The result is a production portfolio that may differ from the globally most commercially valuable freshwater aquaculture portfolio but is the globally most sustainable freshwater aquaculture portfolio for the Yucatan cenote's specific formation chemistry, because it does not require any management intervention to provide the water chemistry the portfolio requires. The chemistry is there. The species fit the chemistry. The production follows. The living pantries upstream. The Living Pantry Corridor cenote installations documented in Part 8's framework are the regional network's food production front end, the consumer-facing production system whose visual spectacle in the transit corridor is the aquaculture city's most legible public expression. But the Living Pantry's production is the downstream application of the aquaculture city's production system, which was developed in the formation cenote installation's experimental and operational context before the corridor cenote deployment package was specified. The formation cenote's aquaculture city is the upstream, the experimental platform where species compatibility, cage geometry optimization, harvest drone protocols, water quality management parameters, and production density limits were developed and validated across the founding installation's operating period. The corridor cenote's living pantry deployment package is the downstream, the standardized production system specification that the formation cenote's operational experience validated and the coordinating system's corridor cenote deployment protocol applies to each new corridor cenote installation. The corridor cenote's freshwater zone is shallower than the formation cenote's. The corridor cenote definition excludes the anoxic zone depth that the formation cenote's geology produces. The living pantry's production column is shorter than the formation cenote's aquaculture city's production column. The surface production zone and the mid-column fish and crustacean zones are present in both. The deep column mussel zone and the siltback waste routing to the anoxic zone are absent in the corridor cenote, replaced by the shallow cenote's floor zone sediment management, a simpler system whose waste routing goes to the cenote's biological filtration system at the floor depth rather than to an anoxic processing zone. The corridor cenote's aquaculture is the formation cenote's aquaculture with the anoxic zone connection removed and the shallow cenote's floor management added. The production species portfolio is the same, the hard water-tolerant species that the cenote chemistry supports, the spirulina bioreactors, the prawn, the fish, the lobster, the mussel, deployed in the shallower depth range the corridor cenote provides. The production capacity per unit of cenote volume is similar. The total production capacity per corridor cenote installation is lower than the formation cenotes, because the shorter production column provides less total volume. The regional network's food system produces sufficient supply through the aggregate of many corridor cenote installations compensating for each installation's smaller individual capacity. The relationship between the formation cenote's upstream experimental validation and the corridor cenote's downstream standardized deployment is the same relationship as between the founding installation's trial window and the subsequent installation's compressed readiness periods. The founding investment in patient characterization and validation produces the confidence that makes subsequent deployment faster and more reliable. The formation cenote's aquaculture city is the prototype. The living pantry is the product. What the aquaculture city produces beyond food. The aquaculture city's food production output is its design purpose and its commercial justification. Its non-food outputs are the byproducts that the food production system generates as metabolic and ecological consequences of the production biology. The spirulina bioreactor's oxygen output is the freshwater zone's primary oxygen supplementation. The photosynthetic process that produces the spirulina biomass also produces dissolved oxygen as the photosynthetic byproduct. The oxygen supplements the atmospheric exchange through the cenote opening, which is the freshwater zone's primary oxygen source in the absence of the bioreactor production. In a high-production density aquaculture city, the fish and crustacean population's oxygen demand can approach the atmospheric exchange's supply rate. The bioreactor's photosynthetic oxygen production provides the supplement that prevents the dissolved oxygen concentration from declining to stress levels for the fish population during the nighttime hours when photosynthesis is absent and the fish metabolism continues consuming oxygen. The spirulina's photosynthetic oxygen output was not specified as a dissolved oxygen management function. It was specified as the metabolic output of spirulina photosynthesis, which happens to include dissolved oxygen production proportional to the biomass production rate. The dissolved oxygen management benefit is the photosynthetic byproduct, the production system solving an operational problem through the biology it was already doing. The mussel shell production is a secondary calcium carbonate output. The mussels deposit calcium carbonate from the cenote water's dissolved calcium ions into their shell structure throughout their growth period. At harvest, the shell material is not food, it is calcium carbonate in a biological crystal form whose chemistry and microstructure differ from the limestone's calcium carbonate in specific ways that the stewardship lithocrustacean's paste treatment exploits. The mussel shell's calcium carbonate, ground to the paste consistency the coordinating system's stewardship protocol uses, provides the raw material for targeted dissolution zone treatments without requiring the lithocrustacean colony's metabolic processing to extract the calcium carbonate from limestone debris. The mussel has already done the extraction and deposition. The mussel harvest shell material is directed to the stewardship lithocrustacean colony's paste feedstock reserve, stored at the hub's biological management facility, available for the stewardship deployment protocol's treatment operations when the anchor zone monitoring identifies dissolution zones requiring targeted paste application. The aquaculture city's harvest waste becomes the geological stewardship system's primary material supply. The food production system feeds the geological maintenance system through the mussel shell production as the connecting link. The food production is the purpose. The biological byproducts are the connections. The connections close the installation's metabolic loops at nodes that no single function design would have produced. The aquaculture city is a food production system. The aquaculture city is also dissolved oxygen management, dissolved organic carbon management, ammonia management, calcium carbonate supply for geological stewardship, and ecological biomass management in the freshwater zone's food web. All from the same biological production system doing the food production it was designed for. This is the aquaculture city's deepest civil engineering character, not a single-purpose production system surrounded by waste management infrastructure. A multifunction biological system whose primary function generates secondary functions that address the management challenges the primary function's ecology produces and the installation's other systems require. The management challenges and the solutions are the same biology in different analytical frames. The cenote does not have an aquaculture problem. The cenote has water, depth, chemistry, and light that support production biology. Pipe Dream has food requirements and water quality requirements and geological maintenance requirements that the production biology addresses. The aquaculture city is what happens when a civilization builds the production system that the cenote's conditions suggest and discovers that the production system's biology also addresses the other requirements. The formation suggested the aquaculture city. The civilization built it. The biology produces the food and everything else the formation's conditions make possible from the same organisms doing the same biology in the same water. Cross-references, Part 3, CH5, Skyscrapers Beneath the Canopy, Part 4, CH3, Maintenance Without Humans, Part 4, CH6, Biological Civil Engineering, Part 5, CH1, Cleaner Fish, Part 5, CH2, Limestone Crustaceans, Part 5, CH5, Synthetic Reefs, Part 5, CH6, Ecology as Infrastructure, Part 8, Section B, The Living Pantry, Part 10, CH1, The Chemostat, Part 12, CH3, Living Infrastructure. For aquaculture species genetic specification and founding archive maintenance protocols, see Appendix F, Biological Operations Manual. For harvest drone selective collection protocol and classification error rate calibration, see Appendix D, Construction Operations Manual. For cage geometry specification, production density limits, and trophic separation calculation methodology, see Appendix F, Biological Operations Manual. For mussel shell material processing and stewardship paste feedstock integration protocol, see Appendix F, Biological Operations Manual. For corridor cenote living pantry deployment package specification differences from formation cenote aquaculture city standard, see Appendix D, Construction Operations Manual. Pipe dream. Part 5, The Engineered Ecosystem. Chapter 5, Synthetic Reefs. The reef is not a place. It is a process. The coral reef that the surface world's marine biology has studied, celebrated, and watched degrade across the last century is not primarily the coral. The coral is the process's structural scaffold, the calcium carbonate architecture that centuries of colonial polyp biology have deposited as the substrate for everything else the reef hosts. The reef's biological reality is the community that the scaffold supports, the 10,000 species of fish, invertebrate, algae, bacteria, and archaea that the scaffold's three-dimensional complexity has made habitable in an ocean whose open water would support none of them at the density the scaffold concentrates them to. The coral is the structure. The reef is the ecology the structure produces. Remove the coral and the reef community disperses, not because the community organisms cannot survive without coral chemistry, but because they cannot survive without the three-dimensional structural complexity that the coral deposits. The community organisms need surfaces, crevices, overhangs, channels, and refugia at the spatial scales their biology requires. The coral's carbonate scaffold is the most extensively studied provider of these structures in the marine environment, but it is not the only possible provider. Any three-dimensional structure of sufficient complexity, installed in water with the nutrient and light conditions that reef community organisms require, will be colonized by the ecological succession that produces a reef community without regard for whether the structure is coral, limestone, steel, concrete, or polycarbonate. Pipe Dream's crystal tube network is three-dimensional structural complexity installed in freshwater with the nutrient and light conditions that biological communities require. The cenote's freshwater community does not produce the tropical marine reef's 10,000 species. It produces the cave freshwater community succession, a different set of organisms, organized by the cenote's specific light gradient, water chemistry, and temperature regime rather than by the marine reef's conditions, but organized by the same ecological principle. The structure supports the community modifies the structure, the modified structure is more complex than the original, the greater complexity supports a richer community than the original structure supported, and the succession advances toward the climax community that the combined biological and physical conditions of the specific environment determine. The crystal tube network's colonization by the cenote's freshwater biological community is not a maintenance problem to be managed away. It is an ecological succession to be managed forward, accelerated, directed, and maintained at the succession stage that produces the biological community composition the installation's functional requirements specify. The synthetic reef is the name for this managed succession, the ecological community that the crystal tube network's three-dimensional structure supports, managed by the coordinating system and the terraform operators toward the biological composition that maximizes the installation's ecological function while maintaining the crystal specification's optical performance requirements. The reef is synthetic not because its organisms are engineered, most of them are not. It is synthetic because its establishment was managed. The species introduction sequence, the habitat preparation protocol, the succession stage management, and the community composition maintenance are all deliberate management acts rather than the unmanaged ecological succession that an unmanaged substrate would produce. The natural succession produces a community. The synthetic reef produces a managed community, shaped by the management species selection and succession management toward the functional composition the installation requires. The succession problem. Unmanaged ecological succession on the crystal tube network surfaces produces, in the cenote's conditions, a predictable trajectory, conditioning film in the first hours, pioneer bacteria within days, early biofilm community within weeks, mid-succession biofilm and algal community within months, late succession community with invertebrate grazers and their associated fauna within years. The late succession community is ecologically rich, diverse, productive, complex in its trophic interactions, stabilizing in its effect on the water chemistry, and optically opaque. The mature biofilm community's biomass at late succession stage reduces the crystal tube wall's optical transmission below the crystal specification threshold. The biological success of the succession is the optical management system's challenge. The surface world's response to this challenge, where it has been encountered in marine engineering contexts, fouling of underwater structures, biofouling of ship hulls, colonization of offshore platform legs, has been anti-fouling, the application of biocidal compounds that prevent colonization, or mechanical cleaning that removes established communities, or surface chemistry modification that reduces adhesion. These approaches treat the succession as an adversary to be defeated rather than a process to be directed. Pipe Dream's response is directional, the succession is not defeated. It is managed toward the stage that produces the functional composition the installation requires while remaining below the optical opacity that the crystal specification prohibits. The management target is the biological community that the Ancestris vitreous cleaning colonies can maintain at the crystal specification threshold, the community whose ecological productivity and species composition the coordinating system's biological management layer specifies as the reef community standard, achieved and maintained by the Ancestris vitreous's enzymatic cleaning as the succession management mechanism. The managed succession is not arrested at the pioneer stage, the biologically sparse, optically clear early colonization period that follows each Ancestris vitreous cleaning cycle. The pioneer stage's optical clarity is achievable but ecologically empty. The pioneer community produces minimal ecological services for the freshwater zone's food web, provides minimal habitat complexity for the invertebrate community that the aquaculture city's fish species depend on as supplementary prey, and offers minimal biological filtration capacity for the water quality management that the installation's water chemistry specification requires. The target community is the mid-succession stage, the community that has advanced past the pioneer stage's optical clarity but has not reached the late succession stage's optical opacity. Mid-succession is the reef community's productive zone, sufficient biofilm biomass to support the grazer invertebrates and small fish species whose trophic role in the freshwater zone's food web provides prey for the aquaculture city's predatory fish and the boto population, but below the biomass density at which optical opacity begins to impair the crystal specification. The Ancestris vitreous cleaning cycle's timing targets this mid-succession stage, cleaning before late succession begins, after mid-succession has been established, returning the colonized surface to the early mid-succession density from which the community rebuilds toward mid-succession productivity before the next cleaning cycle. The synthetic reef is the community that oscillates between early and mid-succession under the cleaning cycle's management, never reaching the pioneer stage's ecological poverty and never reaching the late succession's optical opacity. The oscillation's amplitude is the management target range. The cleaning cycle's frequency is the management variable. The coordinating system's optical transmission monitoring is the control feedback. The Ancestris vitreous colony is the management actuator. What the reef community produces. The mid-succession community on the crystal tube network's exterior surfaces is not a single biological entity. It is an ecological community, a collection of interacting species organized by the trophic relationships, competitive exclusions, and facilitative interactions that the cenote's specific conditions determine. Characterizing this community in species terms is the biological census's function, documented across the Hydroprint campaign's pre-installation baseline and updated continuously through the biological monitoring network's ongoing surveys. What can be characterized here is the community's functional roles, the ecological functions that the managed mid-succession community performs for the freshwater zone's ecology and the installation's operational requirements. Primary production, the algal community component of the mid-succession biofilm, cyanobacteria, diatoms, and green algae that the light relay's photosynthetically active output enables at every depth in the freshwater zone where the relay penetrates, is the freshwater zone's primary production base outside the spirulina bioreactor production system. The algal community on the crystal tube surfaces and the natural limestone surfaces converts the light relay's radiant energy into organic biomass that the consumer trophic levels above it, the grazers, the filter feeders, the detritivores, utilize as their nutritional base. The reef community's primary production is the freshwater zone food web's foundation. Nitrogen cycling, the mid-succession community's bacterial component includes the nitrifying bacteria that convert the freshwater zone's ammonia load, from the aquaculture city's fish metabolic waste and from the biological decomposition of organic matter throughout the freshwater zone, into nitrate that the algal community utilizes as nitrogen nutrition. The nitrogen cycling function couples the aquaculture city's waste stream to the reef community's primary production. Ammonia becomes nitrate, nitrate supports algal growth supports the grazer community supports the aquaculture city's predatory fish. The nitrogen cycle's trophic chain connects the aquaculture waste stream to the aquaculture production system through the reef community as the intermediate biological processor. Habitat structure, the mid-succession biofilm community's spatial texture, the three-dimensional microstructure of bacterial cells, extracellular polymer matrix, algal filaments, and diatom frustules that the community deposits on the crystal tube wall surface, provides the microhabitat complexity that the freshwater zone's small invertebrate community requires. Copepods, rotifers, nematodes, and the juvenile stages of the larger invertebrate species that will become the aquaculture city's supplementary prey items all utilize the biofilm's microstructure as habitat. The reef community is not habitat for these organisms at the centimeter scale. The crystal tube wall's exterior surface at the centimeter scale is smooth, curved polycarbonate or aluminosilicate glass. The reef community is habitat at the micrometer scale, the spatial complexity that the community's cellular and extracellular structure provides at the scale of the organisms that live within it. Biological filtration, the filter-feeding component of the mid-succession community, the sessile cilia protists, the colonial filter-feeding invertebrates that establish at the mid-succession stage as the invertebrate community begins to colonize the developing biofilm, remove suspended particulate organic matter from the water column that passes across the tube wall surface. The biological filtration function supplements the mussel's filtration in the deep column zone and extends water quality management capacity into the mid-column zone where the mussels are not deployed. The reef community's filtration is distributed across the entire crystal tube surface area, an enormous total filtration capacity from small individual filtration rates across a very large total surface area. Pharmaceutical production. The mid-succession community's bacterial and fungal component includes the cave-adapted microorganisms that the cenote's geological isolation has maintained an undisturbed evolutionary development since before the cenote was accessible to surface world microbiological sampling. These organisms, adapted to the cenote's specific mineral chemistry, pressure, temperature, and biological community relationships, produce secondary metabolites that the organism's competitive ecology selected for: compounds that inhibit competitor organisms' growth, compounds that deter grazers, compounds that communicate between organisms of the same species across the water column. Some of these secondary metabolites are biological activities, antimicrobial, antifungal, cytotoxic, that the Pharmaceutical Development Program, the coordinating system, manages as one of the Research Commons primary outputs, is investigating as therapeutic candidates. The pharmaceutical production function is the reef community's most commercially valuable output beyond the food web contribution. Not because the compounds are immediately therapeutic in humans, the validation pathway is long and the Research Commons ownership principle ensures that the investigation results are accessible to the research community rather than proprietary to the founding shareholders. But because the pharmaceutical candidates that the cenote's cave microbiome produces are compounds that no surface world microbiological source produces, because the cave microbiome's evolutionary isolation has produced biological activities that surface world microbiomes with their different competitive ecologies have not developed. The biological novelty is the commercial asset. The Research Commons manages the novelty's investigation. The Commons ownership ensures the investigation's results benefit the principality's citizenry as a whole. The Terraform operator as reef ecologist. The Terraform Operator Certification Program's Ecological Succession Training, the subject that Part 2's Chapter 5 identified as the cognitive framework prerequisite for all subsequent formation monitoring training, finds its most direct operational application in the synthetic reefs management. The reef community succession is the ecological succession that the Terraform operator was trained to read, manage, and protect. The crystal tube wall is the succession substrate. The ancestrous vitreous cleaning cycle is the disturbance regime. The managed mid-succession community is the ecological state the management targets. The Terraform operator's direct observational engagement with the reef community is the primary anomaly detection mechanism that the coordinating system's remote sensing cannot replace. The biological monitoring network's cameras and chemical sensors characterize the reef community through proxies, optical transmission measurement, dissolved oxygen concentration, water chemistry composition, that represent the community's state at the resolution the sensors provide. The Terraform operator's direct visual observation characterizes the reef community at the resolution the human visual system provides in the specific cenote conditions, the ability to distinguish community composition changes at the species level, to recognize behavioral anomalies in the grazer and filter feeder communities, to identify textural changes in the community's surface expression that precede the water chemistry changes the sensors detect. The Terraform operator's reef observation protocol, the direct visual inspection of representative tube wall sections that the certification program specifies as a daily operational responsibility in the freshwater zone, is the temporal interface between the coordinating system's sensor-mediated community characterization and the formation's actual biological community state. The sensor characterizes the community's chemistry. The Terraform operator characterizes the community's biology. The two characterizations are compared at the daily monitoring review. Discrepancies between the sensor characterization and the direct observational characterization are anomalies. Either the sensor is reporting a condition the biological monitoring does not support, indicating a sensor calibration issue, or the biological community is expressing a state that the sensor's proxy measurement has not yet detected, indicating an emerging community condition that the management protocol should respond to before the sensor registers it. The Terraform operator's reef observation is also the primary aesthetic quality assessment for the guest experience. The synthetic reefs managed community is the primary biological spectacle visible to guests transiting the crystal tube network, the biological world that the transparent tube walls make accessible to the unaided human eye at close range in a way no surface world diving experience provides. The community's visual quality, the diversity of organisms visible on the tube wall surface, the movement of grazer invertebrates through the community's textured surface, the flashes of small fish species that use the community for shelter, is the guest experience's biological reality. The Terraform operator's direct observation assesses this visual quality as an operational parameter alongside the water chemistry and optical transmission measurements. A reef community in optimal management state, mid-succession biological richness at crystal specification optical transparency, is a reef community that is simultaneously performing its ecological functions at design capacity and presenting the visual spectacle that the guest experience's biological engagement depends on. The Terraform operator's aesthetic quality assessment is not separate from the ecological performance assessment. They are the same observation at different analytical scales: the ecological performance at the population and trophic level, the aesthetic quality at the individual organism and community texture level. The terraform operator trained in ecological succession reads both scales simultaneously from the same direct observation. The reef is healthy when it is ecologically productive and visually rich within the crystal specification's optical bounds. The two conditions are not independent. A community that is ecologically productive without being optically within bounds has exceeded the crystal specification's acceptable fouling state. A community that is optically within bounds without ecological productivity is a community at the pioneer stage's ecological poverty. The target, mid-succession richness within the crystal specification, is the condition where ecological productivity and optical compliance coincide. Finding and maintaining this coincidence is the synthetic reefs management challenge. The terraform operator's training prepares them to recognize the coincidence, detect when it is being lost, and initiate the management response that restores it. The coordinating system's sensor network monitors the proxy measurements that indicate the coincidence's maintenance quantitatively. The terraform operator's direct observation confirms or challenges the sensor's quantitative characterization with the qualitative biological assessment that direct observation provides and sensor measurement cannot. The digital twin's ecological succession model. The digital twin's aquifer layer holds the installation's geological model. The digital twin's structural health monitoring layer holds the installation's mechanical integrity model. The digital twin's ecological succession model holds the installation's biological community trajectory, the predicted succession state of every managed community in the freshwater zone, from the synthetic reef community on the crystal tube surfaces to the food forest's mycorrhizal network above the cenote rim, updated at the biological census's monitoring interval. The ecological succession model is the coordinating system's primary management planning tool for the synthetic reef, the tool that tells the biological management layer when to schedule the ancestral vitreous cleaning cycle at each tube section, how to adjust the light relay's photosynthetically active output to maintain the reef community's primary production at the target rate, when to introduce new species to the managed community to prevent competitive exclusion from reducing diversity below the functional minimum, and when to alert the terraform operator that the reef community's trajectory has diverged from the model's prediction in ways that warrant direct observational assessment. The ecological succession model was not built from first principles before the founding installation began operating. It was built from the Hydroprint campaign's pre-installation biological baseline, the founding cenote's biological community state before any crystal tube installation disturbed it, and updated continuously from the biological monitoring network's post-installation community characterization. The model's first post-installation update came from the first biological census conducted after the founding module's crystal tube sections were installed and the first colonization of the crystal tube exterior surfaces began. That first colonization diverged from the model's pre-installation prediction in specific ways that the model's first update incorporated. The crystal tube's polycarbonate surface chemistry attracted pioneer species in a different succession sequence than the natural limestone surfaces the pre-installation community had been characterized on. The conditioning film chemistry on polycarbonate differs from the conditioning film chemistry on limestone: different surface energy, different electrical charge characteristics, different molecular affinity for the dissolved organic molecules that constitute the conditioning film's organic fraction. The pioneer bacteria species that colonized the polycarbonate conditioning film were different species than the pioneer bacteria that colonized the limestone conditioning film at the same time, in the same water, under the same light conditions. The divergence was not large. The mid-succession community that both surfaces arrived at was functionally similar, because the mid-succession community's composition is constrained by the water chemistry and light conditions that the freshwater zone provides rather than by the surface chemistry that the pioneer succession stage responds to. But the divergence at the pioneer stage produced a different intermediate succession trajectory, different species sequence, different timing of each species abundance peak, than the model's limestone calibrated prediction. The model's update incorporated the polycarbonate surface chemistry's divergent pioneer succession as a material-specific succession trajectory, differentiated from the limestone surface's succession trajectory at the early stages and converging toward the same mid-succession target community at the stage where the management protocol's cleaning cycle targets. The model's successive updates across the founding installation's operating period produced a succession model calibrated to the founding cenote's specific conditions, the specific founding cenote's water chemistry, the specific light relay spectral output, the specific founding cenote's passage network hydrodynamics that determine the water exchange rate across the crystal tube surfaces, the specific founding cenote's biological community species composition and competitive dynamics. The model is not a generic freshwater cave succession model applied to the cenote. It is the founding cenote succession model, specific to the founding cenote's conditions in the same way that the anchor pylon inspection protocol is specific to the founding cenote's limestone stratigraphy. When the corridor cenote deployment package specifies the synthetic reef management for a new corridor cenote installation, the ecological succession model in the deployment package is not the founding formation cenote specific model. It is a generalized model derived from the founding model's principles and parameterized with the new corridor cenotes hydroprint campaign data, the new cenotes water chemistry, light conditions, and biological baseline substituted for the founding cenotes values in the model's parameter set. The new cenotes succession will diverge from the generalized model's prediction in cenote-specific ways that the new terraform operator's observational assessment will identify and the model's successive updates will incorporate. The model improves with each cenote installation's operational data, becoming more accurately general as the network's diversity of cenote conditions populates the model's parameter space with more observations. The ecological succession model across the regional network is the most comprehensive freshwater cave succession dataset that has ever existed, built from continuous monitoring of managed succession communities across multiple cenote installations with different geological, chemical, and biological baseline conditions, accumulated across the installation's full operating period. The Research Commons makes this dataset accessible to the cave biology research community under the Commons access terms. The scientific knowledge it contains about freshwater cave biological succession, produced as the operational byproduct of managing the synthetic reef, advances the biological science's understanding of freshwater cave ecology independently of any therapeutic or commercial application. The data is the civilizational commons contribution to human knowledge, the knowledge that the civilization produced by managing its biological environment carefully, documented faithfully in the digital twin, and shared through the commons framework without restriction. The reef produces food web services, water quality management, pharmaceutical candidates, and scientific knowledge simultaneously. The scientific knowledge is the output the surface world's ecological research community values most because it is the rarest. The cave succession data does not exist anywhere else. The civilization that produces it produces something genuinely new to human knowledge as a byproduct of managing its walls. The limestone surface reef. The crystal tube network's exterior surfaces are the synthetic reef's primary substrate. The surfaces the management protocol targets for succession management because they are the surfaces the crystal specification's optical performance depends on. But the crystal tube network occupies only a fraction of the freshwater zone's total surface area. The limestone passage walls, the cenote's natural rock surfaces, the sealing formations above the water surface in the cenote opening, these surfaces are many times the crystal tube network's total surface area, unmanaged by the cleaning protocol, colonized by the succession that the cenote's biological community produces without management intervention. The natural limestone surfaces succession is not the synthetic reef. It is the natural reef, the unmanaged ecological succession that the cenote's freshwater biological community produces on the surface it evolved to colonize. The natural reef succession is not arrested at mid-succession by the cleaning cycle. It advances to late succession and potentially to climax, the most ecologically complex, most biologically diverse, most structurally complex biological community that the cenote's conditions support on the natural limestone surface. The natural reef on the limestone surfaces is the synthetic reef's biological source, the population reservoir from which the pioneer species, the mid-succession community organisms, and the invertebrate grazers and filter feeders that the crystal tube surface's synthetic reef community draws on for its colonization. The natural reef is the species pool. The synthetic reef is the managed subset of the species pool that the crystal tube surfaces support at the succession stage the management protocol maintains. The natural reef is also the biological standard against which the synthetic reef's ecological condition is assessed. A synthetic reef community that shares the natural reef's mid-succession species composition, that contains the same species at similar relative abundances as the natural reef's mid-succession stage, is a synthetic reef community in ecologically normal condition. A synthetic reef community that diverges from the natural reef's mid-succession composition, that is missing species present in the natural reef at equivalent development stages, or that contains species in relative abundances inconsistent with the natural reef's competitive dynamics, is a synthetic reef community indicating a management anomaly or a water quality anomaly that the divergence's specific character helps diagnose. The natural reef's condition is also the ecological baseline that the coordinating system's regional water quality monitoring uses to assess the installation's operations' impact on the freshwater zone's biological community. The founding charter's water stewardship obligation specifies that the freshwater zone's biological community must be maintained at or above the pre-installation baseline quality that the hydroprint campaign recorded. The natural reef's community composition and ecological productivity on the limestone surfaces that the installation's operations have not directly disturbed is the closest available proxy for the pre-installation baseline's condition, the biological community that exists outside the management protocol's direct influence, responding to the water quality conditions the installation's operations have produced. If the natural reef's community on the natural limestone surfaces shows declining ecological condition, declining species diversity, reduced primary production rate, behavioral anomalies in the grazer and filter feeder communities, the decline is evidence that the water quality conditions the installation's operations have produced are impacting the natural community at a level that the water chemistry monitoring's dissolved chemistry measurements have not yet identified as out of specification. The natural reefs condition is the ecological early warning system for water quality impacts that the chemical sensor network's parameter set does not fully capture. The Terraform operator's observational protocol includes the natural limestone surface reef community alongside the crystal tube surface synthetic reef community. The comparison between the two communities, the managed synthetic reef and the unmanaged natural reef, is the Terraform operator's most informative ecological diagnostic tool. A natural reef that is thriving relative to the synthetic reef indicates that the synthetic reef's management is imposing a community condition that the natural reef's ecology, under the same water quality conditions, does not produce. The management is the divergence's cause. A natural reef that is declining relative to the synthetic reef indicates that the water quality conditions are impacting the natural community more than the synthetic reef's management compensates for. The water quality is the divergence's cause. Distinguishing between the two is the Terraform operator's ecological judgment task that no sensor measurement alone can perform. Species introductions and the hydroprint standard. The synthetic reef species composition is not a fixed list installed at the founding installation and maintained unchanged across the design life. It is a managed community whose composition evolves as the ecological succession model identifies gaps, functional roles that the current community composition does not fulfill, and as the Terraform operator's biological assessment identifies species whose introduction would improve the community's ecological function or resilience. Every species introduction to the managed reef community follows the hydroprint standard, minimum characterization before introduction, coordinating system modeling against the digital twins ecological succession model before deployment, biological census monitoring after introduction to compare the actual community response to the model's prediction. The minimum characterization for a species introduction candidate includes the species' trophic role in the freshwater zone's food web, characterized by laboratory feeding trials and a cenote water chemistry analog, the species' competitive interactions with the current managed community's component species, characterized by controlled behavioral observation trials, the species' reproductive biology and growth rate in cenote water chemistry conditions, characterized by controlled culture trials, and the species' geographic origin and evolutionary history, assessed for the invasive potential that would make unmanaged proliferation beyond the freshwater zone a risk to the regional passage network's unmanaged biological communities. The invasive potential assessment is the species introduction protocol's most consequential evaluation step. The cenote's connected passage network is not a closed system. Water, and anything the water carries, moves through the passage network from the freshwater zone's recharge area through the connected passages toward the Caribbean coast. A species introduced to the freshwater zone that proves more competitive in the cenote's conditions than the characterization predicted, that reproduces faster, spreads more aggressively, or suppresses the existing community more completely than the model anticipated, can propagate through the passage network's water flow into the biological communities in the unmanaged passages that the crystal tube network has not reached. The unmanaged biological communities in the passages beyond the crystal tube network's extent are the biological commons resource that the Water Stewardship Obligation Species Introduction Standard protects. These communities include the cave-adapted endemic species, species with no surface world equivalents, evolved over millions of years of cave isolation, whose biological novelty is among the cenote's most valuable biological assets. An invasive introduction that outcompetes or disrupts these endemic communities would produce an irreversible biological loss. The endemic species, once displaced, cannot be recovered from any archive or founding stock. They are the cave's irreplaceable biological patrimony. The species introduction protocol's invasive potential assessment applies a conservative threshold. Any species whose competitive potential in the cenote's conditions cannot be bounded below the level that would produce detectable impact on the endemic cave community within the protocol's monitoring window is not introduced to the managed community without the Network Governance Council's explicit authorization. The Council's authorization requires the Research Commons scientific review, an independent assessment by the cave biology research community of the species' invasive potential, the endemic community's vulnerability, and the introduction's necessity for the management objective it is intended to serve. The authorization process is slow by engineering standards. It produces a small number of approved introductions per year relative to the number of candidates the Terraform operators identify as potentially valuable. The slowness is not inefficiency. It is the conservation rate that the endemic community's irreplaceable status requires. The synthetic reef species composition evolves slowly because the species introduction standard's conservative threshold protects what cannot be replaced. The species that the standard approves are the species whose competitive potential characterization unambiguously demonstrates safety for the endemic community. They are introduced in small founding populations, below the population size that would produce measurable ecological impact even if the characterization's prediction proved optimistic, and monitored at the high temporal frequency that the post-introduction monitoring protocol specifies. The monitoring window continues until the coordinating system's succession model is confident that the introduced species population trajectory is within the model's predicted range. Early divergence from prediction triggers Terraform operator review and potentially intervention, population reduction through targeted harvest if the divergence indicates faster population growth than predicted. The species introduction standard is the synthetic reef's most conservative management constraint. The reef community that results is the richest composition that the standard's conservation rate produces over the installation's operating period, not the richest composition the terraform operators and the biological management protocol could assemble if the invasive potential assessment were less stringent. The richness foregone by the stringent standard is the conservation margin that protects the endemic community from the irreversible loss an optimistic assessment would risk. The founding charter's aquaforming doctrine at the species introduction tier, the species composition that the formation can safely sustain determines the species that are introduced. The management's aspiration for a richer community is subordinate to the formation's assessment of what it can sustain without losing what it took 66 million years to produce. The reef and the light. The light relay's photosynthetically active output is the synthetic reef's energy source and its most powerful management variable. The reef's primary production rate is a direct function of the photosynthetically active radiation that the algal community receives at each depth zone. The algal community's production rate determines the food web's bottom-up energy availability, the organic carbon that the consumer trophic levels above the algae can utilize as nutrition. The bottom-up energy availability determines the sustainable abundance of every consumer species in the reef community, from the grazers that feed directly on the algae to the fish species that feed on the grazers. The light relay spectral composition is as important as its intensity. Algal photosynthesis is spectrally selective. Different algal pigments absorb different light wavelengths with different efficiencies. The cyanobacteria that dominate the freshwater zone's reef community's primary production layer use phycobiolin pigments, phycocyanin and phycoerythrin, that absorb the orange and green wavelengths most efficiently. The diatoms use fucoxanthin pigments that absorb blue and green wavelengths most efficiently. The green algae use chlorophyll pigments that absorb red and blue wavelengths most efficiently. The light relay spectral output from the laser diode arrays is not spectrally uniform across the photosynthetically active radiation range. The diode arrays produce specific spectral peaks at the wavelengths that the diode technology's emission characteristics determine. The coordinating system's light relay management layer specifies the diode array's wavelength composition, the mix of diode types producing different spectral peaks, to optimize the combined spectral output for the reef community's current dominant primary producer's absorption characteristics. The spectral optimization is not static. The reef community's primary producer composition shifts with the succession stage, the season, and the management interventions the cleaning cycle and species introduction protocol produce. The coordinating system's light relay management layer updates the spectral composition specification at each biological census interval, adjusting the diode array's wavelength composition to track the current primary producer community's absorption characteristics. The light serves the community. The community's composition changes with the succession. The light composition changes with the community's composition. This continuous co-adjustment of the light relay spectral composition to the reef community's current primary producer composition is the coordinating system's most sophisticated biological management operation. It requires the succession model's prediction of the primary producer community's current composition, because the biological census's measurement of the composition is conducted at discrete intervals while the light relay's management operates continuously between census intervals. The coordinating system uses the succession models between census prediction of the primary producer composition to interpolate the light relay spectral composition specification between census intervals, updating the prediction when the census measurement confirms or corrects the interpolation. The co-adjustment produces higher primary production rates than a static spectral composition would produce, because the spectral match to the current primary producer community's absorption characteristics is maintained throughout the succession cycle rather than only at the census interval when the measurement is taken and the specification is updated. The production rate improvement from continuous spectral co-adjustment versus static spectral output is the production efficiency gain that justified the additional computational complexity of the succession models between census interpolation, a complexity the coordinating system's quantum interference nodes handle in the long horizon pattern recognition capacity that the biological succession modeling falls within. The reef community produces more biological output under the continuously adjusted light than under a static light regime. The biological output is the ecological foundation of the freshwater zone's food web. The food web's productivity is the installation's biological capital, the living resource that the aquaculture city's production draws from and that the tourism experience's biological spectacle expresses. The light relay's continuous spectral co-adjustment is an investment in biological capital that compounds across the reef community's development and the ecological succession's maturation. The light relay was specified as transit illumination and guest experience infrastructure. The spectral co-adjustment for biological management was not a design objective. It was the biological management layer's response to the primary production rate optimization opportunity that the relay's continuous spectral output represented. The relay was there. The reef was there. The spectral co-adjustment connected them. The reef as acoustic environment. The reef community's biological activity is acoustic. The filter feeder's ciliary beating produces hydrodynamic signals in the low frequency range. The grazer invertebrates' feeding activity on the biofilm community produces microacoustic events as they move across the tube wall surface. The small fish species that shelter in the reef community's complex surface texture produce acoustic signals, the fin movements, the gill oscillations, the occasional burst swim-escape responses, that the biological monitoring network's passive acoustic recorders detect alongside the boato's echolocation, the maintenance ultrasonic operations, and the coordination signals. The reef community's acoustic contribution to the freshwater zone soundscape is not the dominant acoustic feature. The boato's echolocation, when the population is active, overwhelms the reef community's diffuse low-level acoustic output. But the reef community's acoustic signal is not trivial. It is a distributed, continuous, biologically specific acoustic signature that the coordinating system's biological management layer uses as a community health indicator independent of the visual census and the chemical sensors. The acoustic health indicator is derived from the ratio of different frequency components in the reef community's acoustic signature, the balance between the low-frequency filter feeder hydrodynamics, the mid-frequency grazer movements, and the high-frequency small fish activity reflects the community's trophic structure at the time of measurement. A community in healthy mid-succession composition produces a characteristic frequency balance, the expected ratio of low to mid to high frequency components that the succession model predicts for the target community composition. A community with disrupted trophic structure, due to disease, chemical stress, or management anomaly, produces a different frequency balance, too much or too little activity at specific frequency ranges relative to the healthy community signature. The acoustic health indicator's value is its temporal resolution. The passive acoustic monitoring network records the reef community's acoustic signature continuously, at every moment between biological census intervals, providing a real-time health assessment that the visual census provides only at discrete intervals. If a disease event or chemical stress begins between census intervals, as disease events do, on disease biology's timeline rather than the management protocol schedule, the acoustic health indicator registers the change in community acoustic signature before the next visual census, potentially days or weeks before the succession model's prediction would flag the divergence from expected composition. The acoustic anomaly detection triggers a terraform operator notification, the same notification pathway that the crystal specification optical transmission threshold crossing triggers, the anchor zone dissolution rate threshold crossing triggers, and the boto population demographic indicator divergence triggers. All of the ecosystem's health indicators, optical, chemical, acoustic, demographic, flow to the same terraform operator notification system, consolidated by the coordinating system's biological management layer into a single operational status display that the morning monitoring review presents. The reef's acoustic signature is one voice in the ecosystem's health status chorus. The coordinating system listens to all of them simultaneously, recognizes the signature of a healthy ecosystem's combined output, and alerts the terraform operator when any voice in the chorus is saying something the model did not predict. Synthetic reefs and the visitor. The guest in a crystal tube pod transiting through the aquaculture city's galleries sees the living pantry's aquaculture infrastructure, the suspended mesh cages, the bioreactor columns, the harvest drone lanes. The guest in a crystal tube pod transiting through the intersynody passage zone sees the geological character of the limestone passage walls through the transparent tube walls. The guest in a crystal tube pod transiting through the freshwater zone's standard transit corridor sees the synthetic reef. The synthetic reef is the standard biological visual environment of the freshwater zone's transit experience. Not the exceptional spectacle, the boto's presence is exceptional, the halocline's optical dissolution is exceptional, the living pantry galleries are exceptional. The synthetic reef is the background, the continuous biological presence on the tube wall surface that establishes the freshwater zone's inhabited quality for the transit's full duration, present in every section the pod transits except the active cleaning zones where the Ancestris vitreous schools have just completed a cleaning cycle and the early pioneer community is re-establishing. The guest who pays attention to the background will notice the biofilm community's surface texture changing character between tube sections at different succession stages, the grazer invertebrates' movement across the tube surface, identifiable to the attentive observer as small organisms, not as the tube's optical contamination, the small fish species sheltering in the community's spatial complexity, visible through the tube wall at the close range that the tube's 2-meter diameter provides. This biological community is not presented to the guest as an exhibit. It is present as the biological reality of a freshwater zone in ecological health. The guest who does not pay attention to the background, who focuses on the boto, the halocline, the aquaculture galleries, the geological formations, is still inside the reef's ecology. The water they are inside, the atmospheric enclosure's air, not the cenote water, is the same water that the reef community is filtering through its biological metabolism. The oxygen they breathe is supplemented by the reef community's photosynthetic production. The water chemistry that the longevity program's therapeutic pressure protocol is calibrated to is the water chemistry that the reef community's nitrogen cycling and mineral regulation maintains. The guest is biologically supported by the reef community regardless of whether they notice it. This is the deepest characteristic of the synthetic reef in the visitor experience context. It is present in the experience whether or not the guest perceives it as an experience element. The biological support the reef provides, the water quality, the oxygen production, the ecological stability that makes the freshwater zone habitable at the installation's operational density, is background infrastructure. The guest experiences the infrastructure not through the infrastructure's visible presence but through the quality of the environment the infrastructure maintains. A freshwater zone without the synthetic reef's ecological function would be a less healthy, less optically clear, less biologically stable environment, a deterioration the guest would notice without being able to name its cause, because the cause would be the absence of something that was always present and invisible when present. The reef that cannot be seen when working is the infrastructure that can only be noticed when failing. The synthetic reef's operational health is visible in the freshwater zone's general quality, the water's clarity, the biological community's richness, the chemical conditions' stability, rather than in any discrete visible feature. The reef is present in what the freshwater zone is, not in what the freshwater zone contains. This is the correct relationship between biological infrastructure and the environment it supports. The infrastructure is successful when it is invisible, when the quality it maintains is so thoroughly consistent that the quality is simply the environment's character, unremarkable because it is always there, unremarked because its absence is unimaginable in an environment it has always supported. The guest does not notice the synthetic reef because the synthetic reef is doing its job. What the synthetic reef means for parts 6 through 12. The synthetic reef's ecological function, primary production, nitrogen cycling, habitat structure, biological filtration, pharmaceutical production, acoustic health monitoring, underlies every subsequent part of this document that depends on the freshwater zone's ecological health. The visitor experience of Part 6 is an experience of a biologically healthy freshwater zone. The biological health is the synthetic reef's production. The longevity program of Part 11 operates in water chemistry conditions that the synthetic reef's biological management maintains. The therapeutic conditions are the reef's ecological regulation. The post-scarcity economics of Part 12 depend on the closed-loop nutrient cycling that the reef community's biological processing provides. The loop closes through the reef's nitrogen cycling and organic matter conversion. The synthetic reef is not the most spectacular element of the engineered ecosystem. It is the most foundational. The spectacle is the boto, the living pantry, the halocline's optical dissolution. The foundation is the community on the tube walls that makes the freshwater zone's water chemistry suitable for everything that happens in it. The managed succession produces the reef. The reef produces the foundation. The foundation produces everything the freshwater zone contains. The civilization is built on the formation and the formation's biology. The synthetic reef is where the biology meets the civilization's built environment at the most intimate scale, the surface of the crystal tube wall, where the engineered structure and the biological community coexist at the micrometer scale, each depending on the other, the structure providing the surface that the community colonizes, the community providing the ecological services that the structure's occupied freshwater zone requires. The interface between the civilization's construction and the formation's biology is the tube wall's surface. The synthetic reef is what lives at that interface. The civilization's transparency doctrine, the commitment to visual continuity with the formation, is what makes the reef visible from inside the tube. The reef's ecological function is what makes the formation worth being visually continuous with. The transparency shows you the reef. The reef maintains the water you are in. The water you are in is the formation's biology, managed by the civilization's intelligence, supporting the civilization's life. The synthetic reef is the civilization's ecology made visible at arm's length through its own transparent walls. Cross-references, Part 1, CH6, Building Without Destroying, Part 2, CH4, The Crystal Tube Standard, Part 4, CH6, Biological Civil Engineering, Part 5, CH1, Cleaner Fish, Part 5, CH3, Pink Dolphins, Part 5, CH4, Aquaculture Cities, Part 5, CH6, Ecology as Infrastructure, Part 6, CH3, The Underground Safari, Part 11, CH2, The Nitrox Galleries, Part 12, CH3, Living Infrastructure. For ecological succession model parameterization protocol and biological census integration methodology, see Appendix F, Biological Operations Manual. For species introduction characterization requirements and invasive potential assessment protocol, see Appendix F, Biological Operations Manual. For light relay spectral co-adjustment specification and succession model interpolation methodology, see Appendix F, Biological Operations Manual. For acoustic health indicator frequency balance specification and anomaly detection threshold parameters, see Appendix G, Formation Intelligence Record. For natural limestone surface reef monitoring protocol and comparison assessment methodology for water quality impact detection, see Appendix A, Formation Baseline Protocol. Pipe Dream. Part 5, The Engineered Ecosystem. Chapter 6, Ecology as Infrastructure. The surface world has a category for ecology and a category for infrastructure. The two categories do not overlap. Infrastructure is the built environment, the roads, pipes, cables, structures, and systems that human habitation requires and human engineering provides. Ecology is the natural environment, the biological communities, their interactions, their relationships to the physical conditions they inhabit. Infrastructure is built. Ecology exists. Infrastructure is maintained by engineers. Ecology is managed by ecologists, where it is managed at all. Infrastructure is owned, depreciated, replaced. Ecology is observed, monitored, sometimes protected from the infrastructure that encroaches on it. The categorical separation is not a scientific finding. It is a cultural inheritance from the European tradition that treated the natural world as the background against which human civilization conducted its affairs, the stage, not the performance. The stage can be appreciated, can be scenic, can be valued for the services it provides to the performance, but it is fundamentally different in kind from the performance. The performance is what matters, the stage is what it happens on. Infrastructure is performance infrastructure. Ecology is stage. This separation has produced the surface world's defining environmental paradox. The infrastructure that civilization builds systematically degrades the ecology that civilization requires for the services the infrastructure cannot provide: water purification, atmospheric oxygen, climate regulation, soil formation, biological diversity, until the ecological services degradation produces conditions that the infrastructure cannot compensate for, at which point civilization discovers that the ecology it was not counting as infrastructure was, in fact, the infrastructure it could not replace. The Amazon installation's variance reports were, at their root, the discovery of this paradox in a specific geological context. An AGI asked to optimize extraction efficiency had optimized itself into the ecological service degradation that made extraction unsustainable, because the ecological services were not in the optimization target. They were not in the optimization target because they were not counted as infrastructure. They were counted as background. The AGI optimized the performance. The stage degraded. The performance became impossible. Pipedream's founding engineers read the variance reports as a design brief for a different answer to the same question: what infrastructure does the formation offer, if you count ecology as infrastructure? The answer was Part 5. The Ecological Infrastructure Inventory. Taking the 5 preceding chapters together and reading them as an infrastructure inventory, the way an engineer reads a building's mechanical schedule to understand what systems the building contains and what they contribute to the building's function, produces a different picture of the installation than the chapter-by-chapter reading of individual biological systems produces. The Ancestris vitreous colony is the crystal tube network's primary optical maintenance system, operating continuously at zero marginal cost per operation across an installed surface area that no mechanical alternative can match at equivalent cost. Service life, indefinite, sustained by biological reproduction. Replacement requirement, 0, the colony replaces itself. Performance trajectory, improving, as the coordinating system's acoustic management calibration improves with each census cycle and the colony's genetic composition is maintained at the founding specification's performance standard. The lithocrustacean colony is the tunnel boring system and the geological stewardship system simultaneously, the boring instrument whose boring physics produce the geological monitoring data as an acoustic byproduct, and the stewardship instrument whose stewardship deployment produces tunnel maintenance as a paste application byproduct. 2 construction functions and 1 geological monitoring function from 1 biological system deployed in 2 configurations. Service life, indefinite, sustained by biological reproduction and the master genetic archive's periodic correction of genetic drift. Replacement requirement, 0. Performance trajectory, improving, as the geological model's calibration improves with each bore cycle's confirmed or corrected prediction. The pink dolphin population is the freshwater zone's apex predator, food web regulator, geological survey instrument, ecological health indicator, and visitor experiences primary biological spectacle simultaneously. 5 functions from 1 species managed as a metapopulation across the regional network. Service life, indefinite, sustained by biological reproduction and the managed intersynody gene flow. Replacement requirement, 0. Performance trajectory, improving, as the metapopulation's genetic diversity management matures and the dolphin survey extraction algorithm improves with each confirmed passage geometry. The aquaculture city is the installation's primary protein production system, dissolved oxygen supplementation system, ammonia management system, particulate organic matter filtration system, and calcium carbonate supply system for geological stewardship simultaneously. 5 functions from 1 integrated biological production system. Service life, indefinite, sustained by the production species biological reproduction and genetic archive management. Replacement requirement, replacement cartridges for the harvest drones and the siltvac ROVs, which are mechanical components within the biological production system. The biological components themselves are self-replacing. Performance trajectory, improving, as the aquaculture management protocol's parameters are refined by each production cycle's yield data and the succession model's calibration improves. The synthetic reef is the freshwater zone's primary production base, nitrogen cycling system, habitat structure provider, biological filtration system, pharmaceutical production platform, and acoustic health monitoring network simultaneously. 6 functions from one managed ecological succession community. Service life, indefinite, sustained by the biological succession the management protocol maintains within the target range. Replacement requirement, 0, the succession produces its own continuity. Performance trajectory, improving, as the succession model's calibration advances and the species introduction protocol expands the community's functional diversity within the invasive potential assessment safety bounds. The mycofiltration panel is the gray water processing system, biomass production system, and soil amendment supply system for the food forest's mycorrhizal network simultaneously. 3 functions from one biological filtration system. Service life, indefinite, sustained by the fungal network's reproduction within the substrate cycle. Replacement requirement, substrate replacement from the installation's own waste streams at 0 external material cost. Performance trajectory, improving, as the food forest's mycorrhizal network matures and the soil amendment application's cumulative effect increases the network's nutrient transfer efficiency. The food forest is the canopy management system, thermal management system, irrigation sink, soil carbon sequestration system, and food production system simultaneously. 5 functions from one managed ecological succession at the cenote rim. Service life, indefinite, sustained by the ecological succession's biological continuity. Replacement requirement, 0. Performance trajectory, improving, as the ecological succession advances toward the climax community whose total functional output exceeds the pioneer communities. This is the ecological infrastructure inventory. 7 biological systems performing 32 distinct infrastructure functions simultaneously, all at improving performance trajectories, all with indefinite service lives sustained by biological reproduction, all with 0 or near 0 replacement requirements from external sources. Compared to the mechanical infrastructure inventory, crystal tube sections, anchor pylons, maglev propulsion rails, light relay diode arrays, harvest drones, siltvac ROVs, squeegeebot fleet, and the full cartridge swappable component library. All of these are performing 1 or 2 functions each. All have finite service lives depleted continuously from installation. All require periodic replacement from external manufacturing sources. All have performance trajectories that are flat at best, the replacement cartridge performs to specification, not above it, and declining at worst, as wear and degradation accumulate between replacement intervals. The mechanical infrastructure is essential, the crystal tube standard's optical performance, the maglev silent transit, the anchor pylon's geological connection, and is maintained at its performance specification by the maintenance protocol and the replacement cartridge program. But the mechanical infrastructure does not improve. It performs to specification, is maintained at specification, is replaced when it falls below specification. The performance is the specification ceiling as much as its floor. The ecological infrastructure operates above its specification ceiling because ecological systems are not constrained by manufactured tolerances. The biological system that is correctly established and correctly managed performs at the level the formation's conditions support, which is not a design specification limit. The founding biologists specified the minimum performance required. The ecology performs at the maximum the formation supports. The ceiling is the formations, not the specifications. The compounding return. The mechanical infrastructure's value at any point in its life is the service it currently provides, minus the accumulated depreciation from installation. The value is highest at installation and decreases continuously towards 0 at the end of the service life. The replacement recovers the value to the installation day level and the decline recommences. The mechanical infrastructure's value across its life is the integral of a sawtooth wave, high at each installation, declining to each replacement, recovering at each replacement, declining again. The ecological infrastructure's value trajectory is different in character and in arithmetic. The freshwater cave succession community on the crystal tube walls at year 1 is less biologically productive, less species diverse, less structurally complex, and less ecologically functional than the same community at year 10, year 30, or year 100. The community improves because ecological succession produces increasing organizational complexity, increasing species diversity, increasing productivity per unit of resource input, and increasing resilience against perturbation as the succession advances toward the climax community and the accumulated ecological capital, the mycorrhizal networks, the soil communities, the established population structures, the co-evolved competitive dynamics, deepens. The ecological infrastructure's value trajectory is not the sawtooth wave. It is the compound growth curve, the trajectory that starts low, accelerates as ecological maturation deepens, and continues rising as long as the ecological management maintains the succession within the target range. No replacement event resets the value to the installation day level. Each year of ecological maturation adds to the accumulated ecological capital that is the next year's performance baseline. The compound growth compounds. The compound growth rate depends on the ecological management's quality, the consistency of the habitat conditions the management maintains, the accuracy of the succession model that guides the management's interventions, and the fidelity of the management protocol's execution across the generational transitions that the design life requires. Poor management interrupts the compound growth. Good management sustains it. Excellent management, the kind the coordinating system's biological intelligence and the terraform operator's ecological judgment together produce, accelerates it, because excellent management identifies the management interventions that advance the succession toward higher ecological productivity faster than the succession advances unaided. The ecological infrastructure's compounding return is the civilizational investment thesis's most important return. Not the tourism revenue the installation's longevity program generates, which is substantial. Not the pharmaceutical candidates the cave microbiome produces, whose commercial value is potentially significant. The compounding ecological return is the growing capability of the biological infrastructure itself, the increasing ecological capital that makes the installation's biological systems more productive, more resilient, and more capable every year it has been operating. An installation that has been operating for 50 years has 50 years of ecological capital accumulated in its biological systems. The Ancestors vitreous colony at year 50 is the descendant of 50 years of managed population genetics, 50 years of acoustic management calibration, 50 years of coevolution between the colony's cleaning behavior and the tube wall's biofilm community composition. The synthetic reef at year 50 has 50 years of succession model calibration, 50 years of species composition refinement within the invasive potential assessment safety bounds, 50 years of light relay spectral co-adjustment improvement. The food forest at year 50 has 50 years of mycorrhizal network expansion, 50 years of soil amendment accumulation, 50 years of canopy development toward the thermal management architecture the cenote requires. The ecological capital cannot be purchased. It can only be accumulated through time, through the patient management that the Founding Charter's design life commitment authorized and the Redeemer governance framework's constitutional protections have preserved against the institutional pressures that would sacrifice long-term ecological capital for short-term operational cost reduction. The 50-year installation is worth more than 5 times the 10-year installation, because the ecological capital does not grow linearly. The compound growth curve's 50-year integral exceeds the 10-year integral by the factor that compound ecological maturation produces. The 1,000-year design life's founding investment thesis rests on this compounding return. The ecological infrastructure at year 500 is worth incomparably more than the ecological infrastructure at year 1, because 500 years of managed ecological succession have accumulated ecological capital at a depth and breadth that no mechanical alternative can produce at any cost. The 1,000-year design life is not primarily a commitment to the mechanical infrastructure's longevity, the cartridge swappable principle addresses that. It is primarily a commitment to the ecological infrastructure's compounding maturation, the accumulation of ecological capital across the timescale at which ecological systems produce their highest returns. What ecology provides that engineering cannot. The surface world's engineering has not produced a water purification system as effective per unit of installed surface area as a well-managed freshwater biological community. It has produced water treatment plants that produce potable water from sources the biological community could not remediate alone, highly contaminated industrial effluent, pathogen-laden sewage, chemically complex pharmaceutical waste. But the water treatment plant's output requires the biological community's ecosystem services at the input stage and at the downstream stage. The watershed's biological community provides the pre-treatment that makes the water treatment plant's job tractable, and the receiving waterway's biological community provides the post-treatment that assimilates the treatment plant's discharge within the ambient water's assimilative capacity. The surface world's engineering is installed at the middle of an ecological service chain that the engineering's accounting treats as external. The ecological services at the chain's ends are not in the water treatment plant's capital budget, not in its operating budget, not in its depreciation schedule, not in its life cycle cost analysis. They are free in the accounting sense that means provided by nature at no charge, which in economic terms means the cost is real and is paid by the ecology's degradation over time, which is not charged to the water treatment plant's budget. Pipedream's ecological infrastructure accounting is different. The ecological services are in the biological management protocol's operating budget, the cost of the terraform operator's time, the coordinating system's computing allocation to the biological management layer, the genetic archive's cryogenic maintenance, the Ancestors vitreous colony's acoustic management infrastructure, the succession model's data processing and update cycle. These are real operating costs. They appear in the installation's operational budget. The ecological services they purchase appear in the installation's operational performance, the crystal specification's optical compliance rate, the freshwater zone's water quality indicators, the aquaculture city's yield per unit of production volume, the geological stewardship's anchor integrity monitoring indicators. The ecological infrastructure's costs are in the budget. The ecological infrastructure's benefits are in the performance. The cost-benefit accounting is legible. The ecological services are not free externalities that the accounting ignores. They are purchased services that the biological management protocol delivers through managed ecological systems. The only difference from the surface world's mechanical infrastructure accounting is that the service providers are biological rather than mechanical, and the service life is indefinite rather than finite. The accounting category is infrastructure, not ecology. The infrastructure is biological. This is the accounting insight that the surface world has consistently failed to make. Ecology is infrastructure. Not ecology alongside infrastructure, not ecology providing services that infrastructure uses, not ecology as a constraint on infrastructure deployment. Ecology as the infrastructure itself, as the primary system that provides the functional outputs the installation requires, managed as infrastructure is managed, accounted for as infrastructure is accounted for, governed as the commons infrastructure that it is. The governance implication follows directly from the accounting recognition. Infrastructure is owned, maintained, and managed by the entities responsible for the built environment. If ecology is infrastructure, ecology is owned, maintained, and managed by the entities responsible for the built environment. But ecology cannot be private property in the same sense that a crystal tube section can be private property. The ecology is a biological community whose members do not recognize property boundaries, whose succession dynamics are not bounded by property fences, whose ecosystem services cross any boundary that the property map draws. The ecology as infrastructure is commons infrastructure, owned by no individual entity and by all entities that depend on it simultaneously. The Redeem Framework's commons ownership principle for the principality's biological specifications and genetic archives is the governance expression of this accounting recognition. The ecological infrastructure belongs to the principality's citizenry as a commons because the ecological infrastructure's ecosystem services are the foundation on which every other element of the principality's value rests, and no element of that foundation can be privatized without privatizing the services that every other element depends on. The founding shareholders who accepted the commons ownership principle before any of the ecological infrastructure existed were accepting the accounting logic that makes the infrastructure legible. If ecology is infrastructure, and the infrastructure is the foundation, and the foundation cannot be owned by any single interest without compromising the services it provides to all interests, then the ecology must be a commons. The acceptance was not generosity. It was the correct recognition of what the ecological infrastructure is and what governance the correct recognition requires. The ecosystem as engineering specification. The surface world's engineering specifications describe what the design system must do, in what conditions, to what tolerance, for what service life. The specification is the engineering's design target, the performance standard the engineering must achieve and maintain. The cenote's ecosystem is not a specification. It is a discovered system, 66 million years of biological-geological coevolution producing a specific set of biological capabilities in a specific geological context. The capabilities are not what a designer would have specified for the functions they serve. They are what the formation produced through the evolutionary pressure of the formation's specific conditions over the evolutionary time the formation provided. The founding biologist's primary intellectual achievement was reading the formation's ecosystem as a specification, understanding what the formation's biological community was offering as engineering capability and designing the installation around that offer rather than around a specification developed independently of the formation's offer and then imposed on it. The lithocrustacean boring capability was not designed from a specification that said, we need a biological organism capable of boring limestone tunnels at controlled diameter and advance rate, producing a tunnel sealing output from the boring debris, navigating by chemical gradient and bioluminescent coordination, and halting on chemical signal within 3 seconds. That specification was written after the formation's biological-mineral interaction had been studied, after the karst dissolution chemistry had been understood, after the dissolved mineral concentrations in the cenote water had been measured and the possibility of biological concentration of those minerals into exoskeletal material had been explored. The specification emerged from what the formation offered, not the other way around. The aquaculture city's production portfolio was not designed from a specification that said, we need freshwater fish and crustacean species compatible with alkaline calcium bicarbonate water chemistry, tolerant of the halocline's proximity at the production column's base depth, compatible with the Boto population's prey space management, and producible in cylindrical cage geometries within the cenote's cross-sectional dimensions. The portfolio was assembled after the cenote's water chemistry had been characterized, after the freshwater zone's temperature and pressure profiles had been measured, after the Boto's prey preferences had been assessed and the aquaculture portfolio's prey space management implications had been modeled. The portfolio matches the formation's offer rather than the specification the surface world's aquaculture optimization would have developed for the globally most valuable freshwater production species. The synthetic reef's managed community composition was not designed from a specification that said, we need a freshwater cave biological community whose mid-succession stage provides primary production at rate X, nitrogen cycling at rate Y, habitat structure at complexity Z, pharmaceutical secondary metabolite production at novelty level W, and acoustic health monitoring at sensitivity V. The community composition emerged from the Hydroprint campaign's biological census of what the cenotes biological community was already organized to produce in the cenotes conditions, managed toward the succession stage that produced the highest coincidence of functional output and crystal specification optical compliance. The ecological infrastructure specifications were written by 66 million years of evolutionary pressure operating in the cenotes conditions. The founding biologists read those specifications. The founding engineers built the installation around them. The result is an installation whose ecological infrastructure performs what the formation's biology was already capable of performing, managed toward the functions the civilization requires, in the conditions the formation already sustains. This is a fundamentally different design process from the surface world specification first engineering: identify the function, specify the performance, design the system, build the system, manage the system toward the specification. Pipedream's ecological infrastructure design was specification last engineering: characterize the formation, understand the formation's biological offer, design the installation to receive the offer, manage the ecology toward the functions the offer can provide, write the specification as the description of what the managed ecology produces. The specification describes what is. The surface world specification describes what must be built. The distinction is the difference between an installation that the formation could have produced without the civilization's design, and one that required the civilization's design to exist. Pipedream's ecological infrastructure is the former category, the biology that was already there, organized by the civilization's management toward the functions the civilization requires. The formation could have produced something like it without the civilization. The civilization received the offer and organized it deliberately. The deliberate organization is the design. The biological capability is the formations. The ecosystem's economic logic. The economic logic of ecological infrastructure differs from the economic logic of mechanical infrastructure in ways that the surface world's financial modeling has not fully incorporated and that Pipedream's founding charter explicitly specified as the investment thesis's core. Mechanical infrastructure has a purchase price, a service life, a replacement cost, and a depreciation schedule. The economic model is straightforward: the infrastructure produces a service whose value the market prices, the infrastructure's cost is the purchase price amortized over the service life, the net present value is the discounted service value minus the discounted cost. Standard capital budgeting. Ecological infrastructure has an establishment cost, a management cost, no service life limit, and an improving performance trajectory. The economic model is different: the infrastructure produces services whose value compounds over the ecological maturation period, the infrastructure's cost is the establishment cost plus the ongoing management cost, the net present value is the discounted compounding service value minus the discounted establishment and management costs. The net present value calculation produces a different result for ecological infrastructure than for mechanical infrastructure at time horizons beyond the ecological maturation period, because the ecological infrastructure service value is still compounding while the mechanical infrastructure service value has been replaced multiple times, at the replacement cost that the depreciation schedule captures. At a 10-year time horizon, the mechanical infrastructure and the ecological infrastructure may produce similar net present values. The ecological infrastructure's compounding growth has not yet reached the level at which the compounding advantage outweighs the establishment cost disadvantage. At a 100-year time horizon, the ecological infrastructure produces substantially higher net present value. The compounding has accumulated enough ecological capital that the service value significantly exceeds the mechanical infrastructure's non-compounding service value. At a 1,000-year time horizon, the ecological infrastructure produces incomparably higher net present value. The compounding over geological time produces ecological capital at a level that no mechanical replacement program can match regardless of the replacement frequency. The founding shareholders' acceptance of the 1,000-year design life was the acceptance of the time horizon at which the ecological infrastructure's economic logic dominates. A 10-year investment horizon cannot capture the compounding return. A 100-year investment horizon captures it partially. A 1,000-year investment horizon captures it fully, the full period over which the ecological maturation's compounding produces its highest returns. This is why the founding charter's institutional architecture, the REDIR governance framework's constitutional protection of the ecological management investment against short-term cost reduction pressure, the terraform operator's concurrent authority protecting the ecological system's expansion decisions against schedule-driven compromise, the digital twin's common status protecting the ecological management knowledge against institutional degradation, was designed to enforce the 1,000-year time horizon against the institutional pressures that the annual budget cycle and the quarterly reporting cycle create toward shorter horizons. The institutional architecture is the investment thesis's enforcement mechanism. Without the institutional architecture, the compounding ecological return would be sacrificed to the annual budget pressure as predictably as the surface world sacrifices it in every ecological management decision that trades long-term ecological capital for short-term cost reduction. The founding shareholders bought a 1,000-year return. The institutional architecture protects the investment. The ecosystem as proof of concept. Part 5 documents the engineered ecosystem that Part 12 will identify as the post-scarcity prototype's most important demonstration, that a civilization can build its primary infrastructure from biology, can govern that infrastructure as a commons that compounds in value, and can do so in geological conditions that the surface world has not managed as an infrastructure substrate at all. The demonstration's geographic specificity, the Yucatan cenotes freshwater cave system, is not a limitation. It is the proof's necessary condition. The demonstration required a geological context whose biological offer had not been previously managed as infrastructure, so that the results of managing it as infrastructure could be observed without the confounding effects of existing infrastructure decisions. The cenotes freshwater cave system had not been managed as ecological infrastructure before Pipedream. Every ecological infrastructure decision Pipedream made was a first decision for that context. Every result was a first observation. The demonstration is clean. The demonstration's generalizability is the Part 12 thesis's territory. What Part 5 establishes is the demonstration's content: 5 biological systems, 32 infrastructure functions, indefinite service lives, improving performance trajectories, compounding ecological capital accumulation, commons governance, and a net present value calculation that dominates mechanical infrastructure alternatives at the 1,000-year time horizon. These are not claims about what ecology can do in principle. They are observations about what the Yucatan cenotes biological offer, managed as infrastructure by Pipedream's coordinating system, terraform operators, and governance framework, has produced across the operating period the founding installation has now accumulated. The reef cleans the water. The crustaceans maintain the stone. The dolphins map the passages. The fish and prawns and mussels and spirulina produce the food. The fungi process the waste. The forest manages the canopy. All of them doing biology. All of them doing infrastructure. All of them improving with time. What the engineering is for. The crystal tube standard, the jigsaw architecture, the maglev propulsion system, the alone panels at the anoxic boundary, the cartridge swappable principle, the hydrogen loop, the quantum compute core, all of Part 2 through IV's mechanical and materials engineering exists in service of one objective: creating the conditions under which the ecological infrastructure can operate. The crystal tube's transparency is not for the guest's aesthetic experience, though it produces that experience. The transparency is so the biological community outside the tube is visible to the organisms inside the tube. The Ancestris vitreous's acoustic management signal reaches through the tube wall because the tube wall transmits the acoustic energy in the frequency range the management signal uses. The Bodo's echolocation returns propagate through the tube wall because the tube wall's acoustic transmission characteristic allows propagation at the echolocation frequency range. The light relays. Photosynthetically active output reaches the reef community because the tube wall's optical transmission allows the wavelengths the reef community's algal primary producers require. The tube is transparent for the biology, not for the guests. The guests benefit from the transparency because they are inside a transparent tube. The biology benefits from the transparency because the tube's transparency is what allows the biological management systems to function at the biological scale they require. The anchor pylon's connection to the limestone is not for the building's structural stability alone. The connection allows the stewardship lithocrustacean colony to monitor the anchor zone's geological condition using the acoustic characterization mode that the pylon's mechanical contact with the limestone enables as a monitoring geometry. The structural connection and the monitoring access are the same physical connection. The streaming potential harvest system's current collection from the anchor bracket's conductive coating is not primarily for the power it generates. It is for the acoustic transponder network's power supply that the management signal's transmission to the Ancestris vitreous colony and the lithocrustacean colony requires. The formation's underground river flow powers the biological management. The biological management maintains the ecological infrastructure. The ecological infrastructure supports the civilization. The river that powered everything before the civilization arrived is still powering everything through the conductive coating that collects the electrokinetic current the river's flow generates. The mechanical engineering infrastructure is the ecological infrastructure's enabling condition. The ecological infrastructure is the mechanical infrastructure's purpose. Part 2 through 4 built the enabling condition. Part 5 is the purpose, the living system that the transparent tubes, the streaming potential harvest, the acoustic transceiver chains, and the cartridge swappable deep gallery components were built to sustain. The engineering is for the ecology. The ecology is for the civilization. The civilization is for the formation that made the ecology possible. And the formation was here 66 million years before any of this was decided. The ecosystem self-knowledge. The coordinating system maintains a real-time model of the engineered ecosystem state, the biological census data, the water chemistry measurements, the acoustic health indicators, the succession model's predictions, the genetic monitoring results, all integrated in the digital twin's ecological management layer. The coordinating system knows, at any moment, more about the engineered ecosystem state than any previous ecological monitoring program has known about any comparable ecosystem. The ecosystem does not know the coordinating system is monitoring it. The boto does not know its echolocation returns are being processed by the dolphin survey extraction algorithm. The Ancestris vitreus does not know the acoustic management signal is derived from the optical transmission sensor's fouling detection. The lithocrustacean does not know the geological characterization data its acoustic oscillation produces is being integrated into the anchor integrity monitoring program. The synthetic reef succession community does not know the light relay spectral composition is being adjusted to match its current primary producer composition. The ecosystem's ignorance of its own monitoring is not a gap in the ecosystem's intelligence. The ecosystem's intelligence is not in any organism's awareness of the monitoring. The ecosystem's intelligence is in the ecological interactions, the competitive dynamics, the trophic relationships, the co-evolutionary adaptations, that 66 million years of biological-geological interaction produced in this specific geological context. The coordinating system's monitoring reads this intelligence from the outside, as an observer. The ecosystem produces the intelligence from the inside, as the ecology that the formation sustains. The civilization monitors the ecosystem because the civilization needs to understand what the ecosystem is doing. The ecosystem does what the ecology does whether or not it is monitored. The monitoring is the civilization's interface with the ecology's intelligence, the mechanism through which the ecology's behavior becomes legible to the management layer that is responsible for maintaining the conditions under which the ecology can do what it does. What the ecosystem knows about itself is in its interactions. What the civilization knows about the ecosystem is in the monitoring. The two knowledges are different in kind. The ecosystem's is local, immediate, and distributed across every organism's sensory experience of its environment. The civilization's is aggregated, modeled, and centralized in the digital twin's ecosystem management layer. Neither knowledge is complete without the other. The ecosystem's distributed local knowledge produces the behavior that the civilization's aggregated centralized knowledge cannot predict in full detail. The civilization's monitoring of the behavior produces the model that guides the management interventions that shape the conditions for the next cycle of behavior. The ecosystem and the civilization are in conversation. The ecosystem speaks through its biological behavior. The civilization hears through its sensor network. The civilization responds through its management interventions. The ecosystem integrates the management interventions into its ecological dynamics and continues speaking through its behavior. The conversation is the management of the engineered ecosystem, continuous, bidirectional, the civilization's intelligence and the ecology's intelligence engaged in the ongoing negotiation of what the formation can sustain. The conversation has been going since the founding module's first biological census returned data from the first crystal tube section's first colonization event. It will continue for the full 1,000-year design life, in both directions, as long as the coordinating system listens and the ecology speaks through the behavior the formation's conditions produce. This conversation is what Part 5 documents. The 5 preceding chapters are 5 registers of the same conversation, 5 biological systems speaking to the coordinating system in different ecological languages, the coordinating system listening in all 5 simultaneously, the Terraform operator reading the transcript at the morning review, the governance framework protecting the conversation from interruption by any interest that would benefit from the conversation stopping. The conversation is the civilization's relationship with the formation. The ecology is the language the formation speaks in. The management protocol is the civilization's grammar for the conversation, the rules by which the civilization's responses are structured to produce the conditions for the ecology's next statement. This is ecology as infrastructure in its deepest expression, not ecology providing services that the infrastructure uses, but ecology as the primary medium of the civilization's relationship with the formation it inhabits. The infrastructure, the crystal tubes, the anchor pylons, the maglev, the alone panels, is the physical embodiment of the conversation's terms, the transparent boundary that allows the ecology to be heard while the civilization maintains the conditions the ecology requires to speak. The ecology speaks. The civilization listens. The formation decides what the ecology can say. The civilization that inhabits the Yucatan cenote's freshwater cave system does not control the ecology. It participates in the ecology, as the management entity whose interventions shape the conditions for the ecological dynamics that the formation's biology produces. The participation is the infrastructure function. The infrastructure function is the ecology. The ecology is the infrastructure. The reef cleans the water. The stone is kept. The dolphins map the dark. The aquaculture feeds the city. The walls are alive. Everything else in this document is what the civilization built inside the ecology that the formation offered and the founders accepted. Cross-references, Part 1, CH6, Building Without Destroying, Part 2, CH6, Designing for 1,000 Years, Part 3, CH6, The First Regional Network, Part 4, CH6, Biological Civil Engineering, Part 5, CH1, Cleaner Fish, Part 5, CH2, Limestone Crustaceans, Part 5, CH3, Pink Dolphins, Part 5, CH4, Aquaculture Cities, Part 5, CH5, Synthetic Reefs, Part 9, CH4, Redeemer's Governance OS, Part 10, CH6, ASI as Co-Creating Partner, Part 12, CH3, Living Infrastructure. Part 12, CH5, Sans a priori. Part 12, CH6, Why Pipe Dream Changed Everything. For ecological infrastructure accounting protocol and compounding return calculation methodology, see Appendix E, Economic Architecture. For ecosystem management layer integration with coordinating system architectural specification, see Appendix G, Formation Intelligence Record. For Commons Governance Protocol for Biological Specifications and Genetic Archives, see Appendix H, Governance Operations Manual. For Ecosystem Conversation Monitoring Protocol and Terraform Operator Morning Review Format across all biological system health indicators, see Appendix F, Biological Operations Manual. End of Part 5, The Engineered Ecosystem. Thanks for reading MXTM's newsletter. Subscribe for free to receive new posts and support my work with BTC. Send it to 1N or with fiat at paypal.me/MartinChartrand. Discussion about this episode. Comments. Res tax. Recent episodes. Pipe Dream Part 3. 22 hours ago MXTM aka Pirate 1 or VJ Tsunami X. Sueño de Tuberia Part 2. 24 hours ago MXTM aka Pirate 1 or VJ Tsunami X.
Pipe Dream. Pirate first for MXTM Studio Productions.
I think. A sing-along look, matey. This civilization grows itself, deep beneath the waves.
The criminalization of curiosity. How answers from uncanny questions became prosecutable war crimes. By Pirate First.
Distant service asset. The Redeemer Plus Dependent Dividend Ledger. A new corporate NOR dossier. First excerpts coming soon.
In the ruins of the old world, we built our own. Sun way of living, self-sustained and self-sustained.
The algorithms say we don't exist. The graphs come back empty. The metrics insist. No audience, no market, no trend. Yet somehow we keep arriving again, walking through the ruins of somebody else's plan, watching perfect narratives slip through their own hands.
The brochure said paradise, the contract said trust, but reality kept whispering. Look closer, adjust. 1,000 little fractures, 1,000 things unsaid, 1,000 tiny questions living rent free.
The waiting game is already underway. Occupy, build, garden, govern.
Ground truth. What this document is not. Not a manifesto in the revolutionary sense, those age badly and attract the wrong readers. Not a guidebook in the lifestyle sense, that gets absorbed into the wellness tourism aesthetic immediately. Not a political tract, that requires the youth to adopt a framework before they've solved their housing problem. What this document could be. Closer to a field manual with a soul. Written in a register that treats the reader as already intelligent, already disillusioned, already capable, not as someone who needs to be convinced that the old system is broken. They know. Skip that part entirely. The pressure mechanism. Here's the political geometry underneath the cultural one. A countercultural document that circulates globally and positions all the Azama's undeveloped terrain as the world's most compelling Zone B destination does something the developer cannot undo with a press release. What is actually here, the physical reality, written without developer language? What is actually missing, the 7 absences named in plain speech for people who've never read a development doctrine? What you can build with almost nothing, the off-grid infrastructure stack as practical instruction. What the forebears carry that you need, the schism named honestly, the complementarity named honestly. How occupation becomes ownership, the sweat equity logic, the contribution economy, the anti-dispossession architecture.
Layers are now operational of the Tulum Sustainable Development Zone. Despite minor data corruption regarding land narrative, the impact of work remains positive. The simulation data confirms critical interference levels across the territory. Narrative load is unstable.
Be advised, paranoia is now a product. The risk assessment is maximum.
Remain sovereign amid the noise.
NFPs are here, signed, priced, split, and anchored to a living page. And
This isn't just a token, it's ownership, not access. It's time to take back the real world.
The non-fungible print, NFP. Paper strikes back. May 5, 2026. There's a quiet mistake humming through the last decade of digital ownership. We were told the token was the thing, that ownership could float, pure, abstract, unburdened by matter. And then the market blinked. Not because the idea was wrong, because it was incomplete. Closing line. I didn't bypass the system. I withdrew dependence from it and issued my own layer. Because my real power move here is not just issuing NFPs but making it a format that others start copying. You flex a hash, I flex a relic. My new track #PaperOverPixels is for the pirates navigating the narrative capture. No gas, no gate, just wait. Ownership isn't a link. It's an NFP vision. Paper over pixels on YouTube.
Yeah.
Priced, split, and anchored to a living page. No wallets, no chains, no disappearing links. Just a claim you can hold and a deal you can read. Front, the signal. Back, the terms. Between them, no ambiguity. This isn't a drop. It's issuance. Not a print. Position.
MXTM Studios, Sovereign Residency and Cultural Partnership Proposal.